We do mostly, I think, have found this, but we have a light breakfast fare and coffee, tea, drinks over towards stage right, or my right, your left. And then I just want to talk about many of the resources and then the contact information of our presenters today. Anything that's mentioned in today's program is going to be posted on the University Research Symposium website within a week of this event. You can find that website very easily by typing in go.ncsu.edu/research-symposium. And registrants will get a reminder of this when resources are all posted. And then I just want to encourage you all, take this opportunity to meet with your researchers and administrators here at NC State. The speaking portion begins at 10 a.m., so you'll have a couple more minutes to mingle with and meet people in person that maybe you haven't seen in a couple years. Last, maybe housekeeping note, in the back, you'll see in the back two corners, we do have what we're calling charging stations, so if you want to keep your laptop charged, we have a couple of areas in the back that you can plug that in and be ready for anything you might have to grab on your laptop. So charging stations in the back. And we'll start shortly. Thank you.
Good morning, everyone. All right, we're ready to get going today. Dr. Mladen Vogt, our Vice Chancellor for Research, is unable to be here today, but he sends his regards. So I'm Genevieve Garland, Associate Vice Chancellor for Research, and so I'm here to welcome you all today. So we want to thank you for joining us for the 2024 University Research Symposium. And as many of you know, every year we host, along with the Research Leadership Academy, the University Research Symposium. And it's a cross-disciplinary research symposium here at NC State. And this year, the topic is 21st Century Threats to Health. The goal of this symposium really is to bring together researchers and scholars to talk to and meet with each other and for them to explore opportunities for new partnerships and envision the future of research here at NC State. We encourage you to make connections and expand your knowledge of research being conducted here at our own institution. And what a great topic we have here today. As we know, we have lots of expertise crossing animal health, human health, agriculture, and many other topics as well. I'd like to start off today by inviting Dr. Heike Sederoff to the stage. She's a professor and university faculty scholar in the College of Agriculture and Life Sciences, and she's this year's chair of the URS Planning Committee.
Thank you, Genevieve. And good morning, everybody. Still some empty spots, but I think it will fill in pretty nicely today. So I just want to give a couple of sentences. I don't want to take too much of your time. I want to explain why we picked this topic, 21st Century Threats to Human Health. This topic came from a theme. The theme came from an announcement of a recent formation of the Lancet Commission on 21st Century Global Health Threats by the United Nations Population Fund and the Institute for Health Metrics and Evaluation and many others. And I want to read a little bit, just a paragraph, I know it's early, from this announcement of the formation of this committee. And that says the 3 C, and I cite, the 3 Cs, COVID-19, climate change, that's 2, and conflict, as in war, highlight that the steady global health progress of the last 70 years will not necessarily continue into the next 70 years. But there are many other threats beyond the 3 Cs that threaten to undermine our future global health progress, including rising antimicrobial resistance, increasing obesity, inverted population pyramids, eroding sexual and reproductive rights for women, food insecurity, and the frailty of multilateralism. So many of these threats interact. They are cause and consequence at the same time. And they lead to complex health and economic, social, and geopolitical consequences. So our food systems specifically can be designed to improve health, reduce poverty, and slow climate change, or a bad design will do the opposite and will increase these things. Cross-cutting technologies and policies are therefore really needed to reduce the risk and emerging threats. So we have a really what we invited, the people we invited to the stage today, and we have highlighted speakers. We have quick lightning rounds of short presentations, and we have panels to grapple with the whole breadth of this and all the researchers, or some of the researchers that we have at NC State that deal with that, you guys. I want to start off with introducing the first 3 speakers, 3 highlighted speakers in our first panel. And Jacob, if I can invite you up. Dr. Jacob Jones is a Kobe Steele Distinguished Professor of Materials Science and Engineering. He's also the director of the NSF Science and Technology for Phosphorus Sustainability and Short Steps Center, and the director and PI of the Research Triangle Nanotechnology Network. And Jacob will present his research today, and I think some of the STEP Center research on 3 phosphorus-related threats to 21st century human health. Thank you, Jacob.
Anyone watch that basketball game on the weekend? Go Wolfpack. All right, so I have about 15 minutes, and I want to save some time for Q&A, which I think is very valuable. And in no way in 15 minutes can you introduce both the threats and the real-world solutions. So I hope what I can do in the spirit of the topic of the seminar is present 3 explicit phosphorus-related threats, and then give you the opportunity to bring your big ideas and create innovative solutions to some of these challenges that we pose. So I think first and foremost, we need to acknowledge that phosphorus is important. It's an essential nutrient in our food system for animals, plants, and microbes. It facilitates cell development. It's a key component of molecules that store energy, store and process genetic information, and form cell membranes. In fact, about 85% of the phosphorus that we intake and store in our bodies is stored in our bones and our teeth. And therefore, the recommended daily allowance from NIH of phosphorus for children is over 1 gram, and for adults is about 700 milligrams. But the way that we use phosphorus is unsustainable, and it's harmful to the environment. On the bottom, you can see we source most of the phosphorus we use in the food system from mined locations from non-renewable resources. Phosphorus is an essential nutrient in the food system, but it's also a pollutant. And only 20% of the phosphorus that enters the food system actually enters our diet. So 80% is wasted. It's an extremely inefficient process. And ultimately, a little bit of that phosphorus that seeps into, runs off into, or is an effluent from wastewater treatment plants causes eutrophication and associated algal blooms and fish kills. There we go. Importantly, from a temporal perspective, as a society, we've increased our dependence on this non-renewable resource tenfold since around the 1950s. It was the first and only president that actually uttered the words phosphorus in public, was FDR back in 1938, and he said he cannot overemphasize the importance of phosphorus, not only to agriculture and soil conservation, but also to the physical health and economic security of the people of the nation. And so this is a topic that very much resonates with the topic of this particular symposium. Shortly thereafter, during the baby boom, we've used phosphorus as well as other contributions to increase the productivity and crop yields in our food systems in order to feed the growing population of the U.S. You can see, for example, on the right, the crop yields of corn as one example, starting around the 1950s, just increasing steadily. And again, this is a necessary nutrient for a food system in order to feed us as humans. And so the first threat that I want to introduce is supply chain risks and price volatility, and the fact that it undermines our food security moving forward. As we noted, most of the phosphorus used in fertilizers is sourced from mines, and fortunately here in the U.S., we mine most of our phosphate domestically, and those mines are located predominantly in Florida and North Carolina. That's not the case around the world. These phosphate deposits are disproportionately distributed throughout the world, and there are some countries that always have to import their phosphorus supplies, including countries that will become major agricultural producers in the coming decades. On the right, I pulled some data from the USGS, which shows both the phosphate mined in 2022 as well as the reserves as a function of various locations in the world. You can see in the US, we mine about 21 million. These numbers are in, I think, thousands of metric tons. We mine about 21 million of metric tons of phosphate every year. And we have about 1 billion tons in reserve. And you do the calculation, maybe we have about 50 years before it's exhausted. So the next generation, our children and our nieces and nephews, may see a different world. If you look at other places in the world like Morocco, Morocco holds the world's largest reserves of phosphate, and this is a very significant resource as we think about the future. I'd also point out here, Russia at the bottom is a major producer, as well as China, and we'll see their influence play out on the next slide, which shows us the price for fertilizers. As one example, we show diammonium phosphate as one representative fertilizer and the monthly price as a function of time. You probably can't see the dates at the bottom, but I've identified a couple dates at the top of relevance. In 2008, there was a significant price spike. That is an order of magnitude increase in price. This had dramatic consequences for certain agricultural producers, in particular small farms. And then the price spike subsided, but then around 2021 and 2022, with certain things going on in the world, the price once again goes back up. So this is something of serious consideration when thinking about food security of the nation and food security overall for the world. Threat number 2 is that excess phosphorus leads to eutrophication and harmful algal blooms. I mentioned this briefly before, but there are some more details provided on this slide. So eutrophication occurs when the environment becomes enriched with nutrients. It was back in the late 1960s and early 1970s that folks were doing so-called whole lake experiments in Canada, testing out the influence of these different nutrients on eutrophication. And this image shown here is really important in the field of limnology and in phosphorus sustainability, because it shows very dramatically that on one side of a curtained lake where it's dosed with nitrogen and organic carbon versus the other side where it has the same nitrogen and organic carbon but additionally dosed with phosphorus, you can see eutrophication occurring. And so it was from these types of experiments that everyone accepted that phosphorus is the major driver for inland eutrophication, whereas nitrogen is the driver for red tide, for example. This resulted in phosphate bans in detergents in the 1970s, but phosphate bans in detergents is just one aspect of a solution. A couple other major contributors to this consequence is that phosphorus is insufficiently extracted from wastewater, and it runs off from certain agricultural lands. Now, this causes not just challenges to the environment, but it's an environmental and economic consequence, in particular in North Carolina, if we think about coastal communities and the impact that this may have to fisheries, for example. It's not just a local problem, it's a global problem. Now, this graph is a bit dated, and so I think the situation is actually worse than is presented here. But what this graph shows is the water pollution level resulting from phosphorus in inland waters. And colors of orange and red are bad, and you can see that throughout North America, we have challenging water pollution problems, and also through other areas of the world, like Europe, for example, and Asia and Australia. The third threat is growing radioactive phosphogypsum waste from fertilizer production. And in the image in the background here, you're looking at a view of one of these phosphogypsum stacks, which has toxic water that contains phosphorus and nitrogen in the middle. So to produce fertilizers from phosphate rock, if there's any chemists in the room, you'll feel satisfied by looking at a chemical reaction. You take something like apatite and you react it with sulfuric acid, and this forms phosphoric acid, which is used as a fertilizer, but there's also a byproduct, which I've outlined there in red, which is calcium sulfate hydrate. This is phosphogypsum. And phosphogypsum contains radioactive elements. The reason is because the source, apatite, contains a non-negligible amount of radioactive elements, and they end up in this solid byproduct, which is phosphogypsum. So they're not extracted through that process. As a result, most of the phosphogypsum that results from beneficiation of phosphate rock into phosphate fertilizers, most of it ends up being unused for any other purpose and stored in stacks indefinitely, known as phosphogypsum stacks. Another photograph which is shown here in the upper right. So it's interesting to note a couple of policy things associated with this. So you'll note the dates in the lower left can be assigned to two different White House administrations. And the first one, in October of 2020, the EPA approved the use of phosphogypsum in government and construction projects. And then the EPA went back on that in June and withdrew that approval. So there are policy issues, there are environmental issues, there are human health issues. Those familiar with phosphogypsum stacks may also remember that back in April of 2021, there was a scare in Florida with one of the largest phosphogypsum stacks. There was a breach, a small leak coming through this stack, and the toxic water that was on the top was leaking out. And of course, you immediately have concerns that the leak is going to increase in size, and eventually there may be a collapse of one of the walls. And there were predictions at the time that 10 feet high wall of water would rush over communities in the area when the stack fails. And therefore, there was declared a state of emergency, and folks came in order to rapidly pump out the water, and there's no place to put it other than Tampa Bay. And so in the lower right, you can see the release of some of those toxins predicted in models in Tampa Bay, which obviously has consequences for eutrophication and algal blooms. So all this negativity aside, What can we do toward phosphorus sustainability? So I'll tell you two things. The first relates to research. It's impossible to unpack the research going on in STEPS in this short amount of time. But what I wanted to put forward to this particular audience is a roadmap that we helped create with RTI International. There are many roadmaps out there for phosphorus sustainability. Most of them are European-centric, and in Europe they have a different perspective on environmental conservation, for example. This one was informed from US perspectives, and it presents 9 impact opportunities. At the very top, increased awareness. Before walking in here, I'm not sure how many of you knew that there were challenges with phosphorus sustainability, but what you can do, and this is emphasized on a subsequent slide, is you can help us amplify the message, amplify the challenges and some of the solutions that individuals can help us with on a day-to-day basis. But then lower, somewhere down here in the middle of the bottom, is reduce mining waste. Right? So how can we think about that beneficiation process, or think about other chemical processes where we could transform the phosphogypsum into something that's usable and non-radioactive? And there's a host of other things. So I encourage you, if you're interested in contributing research in this area, to download the roadmap and see some of these targeted opportunities that collectively the community has elevated. And then the other thing is what you can do as an individual. STEPS is a partner with SPA, the Sustainable Phosphorus Alliance, and they produce something that I think is really exceptional. I think they produced this about a year ago. It's 12 steps to a more phosphorus efficient you. So say that 5 times fast. And so here, think about the fertilizers that you apply to your lawn. As soon as I saw this, I stopped applying fertilizer to my lawn because it runs off into waterways immediately. There are some other things you can do, whether it's outside or inside. You can choose phosphate. You can choose detergents that do not contain phosphate. I was surprised when I read the ingredients of the car wash that I have, which actually still contains phosphates. So you can think about this, and you can take actions as an individual that would help contribute. And then finally, I want to highlight on April 4th, we're going to have a visiting speaker, Jim Elser. Actually, on April 4th, he's visiting STEPS, and on April 5th, he's visiting the Department of Applied Ecology. He's giving 2 talks back to back. The one on April 4th is open if you'd like to hear more details about the challenges underpinning phosphorus sustainability and some of the solutions. We would invite you to come to the seminar in person in Fitzwillard Hall or join over Zoom. And then on the right, what we've done in order to increase awareness is a few organizations that are interested in phosphorus sustainability have created a week dedicated to phosphorus sustainability. We call it Phosphorus Week. It's the 15th week of the year in celebration of the 15th element on the periodic table. And so on Phosphorus Week, what we'd invite you to do is help us amplify on social media some of the challenges that we have. Help us amplify some of the successes that the scholars in the center are achieving, some of the innovative results that they're achieving, and set an example, set a personal example. And with that, I finish. Thank you.
Questions? It's too early? It's Monday? And I guess I should have said, I would like to use we do have a live stream going, so I'm going to try to use the microphone for questions.
Hi, really excellent snapshot there. My name is Jack Odell. I'm a professor in the Department of Animal Science, and I just want to give a comment that, I don't know, maybe in the last 15 years or so, there's been really concerted effort in animal nutrition and the use of phytase as an enzyme, because so much of the plant-based phosphorus is not bioavailable and consumed by humans or animals or whatever. It's phytic acid, and they've engineered these phytase, microbial-derived phytases to really help in that dimension of this very complicated schema here or whatever, and that's been some recent progress. I think that's really good.
Yeah, absolutely, and thank you for bringing that up. It's not an area that I emphasized here, and in fact, it also speaks to dietary choices, for example, right? So one of the things I didn't have time to fit into the slides is the influence of your dietary choices on the phosphorus footprint right? Your upstream phosphorus footprint. So how much phosphorus did it take to produce the foods that you eat? And that's a serious consideration. I think it's alluded to in the 12 steps to a more phosphorus efficient use, but there's not a lot of information out there. But animal products, for example, because of the food system and because of the need to feed and grow animals, animal products have a lot higher phosphorus footprint than vegetables, for example. Now, I'm not advocating everyone become a vegan. We'll see at lunch, people's behaviors and their choices, but also I'd emphasize food waste as something to take into serious consideration, because food that you eat that has a high phosphorus footprint, an upstream phosphorus footprint to create it, it's much more disastrous to waste it than wasting a product that had a lower phosphorus footprint. So it touches upon food waste as well. Thank you for bringing it up.
I think this is a follow-up question. Thank you, Jacob. On your steps list here, you mentioned things like microbial and plant-based solutions, circularity and improvement. And so is that like crop rotation and other approaches? And then how do you work with farmers directly to try to help change practices and kind of the big picture?
Yeah, great question. Are you referring to the roadmap?
Yeah, I think so, yeah.
Okay, yeah, there are a number of people in STEPS that are part of the extension program. And I would highlight the work of Luke Gattiboni in particular, who about a year ago, through experiments that were partially supported through STEPS, was able to come up with new recommendations for phosphorus fertilizer use in North Carolina, actually reducing it a little bit. And I think that you're pointing to something that's very critical to a problem this complex, which is stakeholder engagement. And the extension program can put us on the front lines talking with farmers and adopters, and that's very important.
That's okay.
Hi, thank you so much for the talk. What's your comment in regard to organic and inorganic phosphate? And what's the bigger problem, or could you elaborate on that?
Yeah, there's lots of words floating around. So organic, inorganic, bioaccessible, for example. So a lot of the inorganic phosphate that's bioaccessible becomes locked into soils. It becomes mineralized, for example, or there are organophosphate compounds used in herbicides and pesticides which cannot be broken down. I think all of them present unique challenges. At the end of the day, what you need is a bioaccessible form. So the more you can think about what's out there and how to transform it into something that's bioaccessible, then the better you are set to help really address the problem. Okay.
Thank you, Jacob. Jacob, I was thinking maybe what would be great to get this out further is if Step Center could generate a list of low phosphate alternative products for households, so we don't all have to go through it. I would like that. Yeah. Thank you. Our next speaker is a brand new distinguished William Neal Reynolds, distinguished professor in entomology and plant pathology. She's also the director of Emerging Plant Disease and Global Food Security, in case you see a theme here, cluster. It's Dr. Anna Whitfield. She will present, she works on viruses, emerging viruses and vectors threatening our food security and our wine production. So listen carefully. She just told us that that's a major new thing. Her title is Emerging Plant Diseases at the Intersection of Food Security and Health. Thank you, Anna.
Well, thank you, Heike, and the organizing committee for this opportunity to talk about my research and to highlight the importance of emerging plant diseases and food security. So today, during my talk, I'm going to tell you, really emphasize the importance of plant diseases and plant pests on food security. And then I'm going to give you a case study with my favorite virus. Of course, I'm a plant virologist, so we have favorite viruses, which you can see here. It's going to be tomato spotted wilt virus, which is a really important pathogen in the state of North Carolina and globally. So I know people in this room are very familiar with this slide, that the global population is increasing. We're going to have 9.3 billion people soon. And that's going to be, there's going to be a significant need for more food, 60% more food than is consumed today. So for those of us working in food production systems, this is an enormous challenge, and it's going to require really integrated multidisciplinary research like we do here at NC State to address this problem. And as highlighted in this quote from Anthony Blinken, the Secretary of State, food security is also a national security issue. And so we really need to be cognizant of that as well, and how food security around the globe is affected. So just to point out that world hunger is on the rise again. So we were dipping around 2015. We saw a decrease. But now in 2022, the most recent number that I was able to find is that more than 900 million people are food insecure. And as those of you who are aware, the global pandemic and now inflation is really contributing to this food insecurity. So we're having more demand on the system. I also want to highlight the burden of nutritional disorders on the healthcare system. And so highlighted here I have stunting and wasting. So this is a really important problem. And I'll just say since 2015, these numbers haven't budged. They haven't decreased. We're really at the same place. We have an enormous amount of poor nutrition. And then we're also seeing the opposite effect in developed areas like the United States, where we have the additional burden of children being overweight. And that's an enormous burden and as significant as the burden as infectious disease, these nutritional disorders. So it's really important that we focus on improving the quantity of food, but also the quality of food that people are consuming. And I just want to point out some really nice data showing the global burden of pathogens and pests on major food crops. So if we're going to increase our food production without taking on more land and more significant inputs, one key target area that we have is to reduce the losses due to pathogens and pests on major food crops. And so here I've highlighted wheat, rice, maize, potato, and soybean. And you can see with the percent yield loss that's here at the bottom. Can you see this pointer? Yeah, so the losses are 20 to 40% in these major food crops. So there's an easy way for us to recoup some of these losses. So I want to highlight the work of our Emerging Plant Disease and Global Food Security Cluster. We're really interested in understanding the fundamental basis of emerging infectious diseases and developing technologies to predict and manage these pests of plants and to generate the next generation of globally engaged scientists. So for my case study today, I'm going to talk about tomato spotted wilt virus. You can see that here in this picture. This is a diseased plant. It's going to have a significantly reduced yield of tomatoes. This virus is distributed throughout the world, and it has a really broad host range and affects over 1,000 different plant species. So it's one of the plant viruses with the largest host range. So that makes it a problem on a lot of important crops like tomato, pepper, peanut, really important food. This virus is vector-borne, and I'll just say vector-borne diseases are a major problem throughout the world. And that's the emphasis of the research that I do, but I can just tell you that this is an ongoing and increasing problem with these vector-borne diseases. And so we see, and I think this is a small estimate, over 1 billion in losses annually due to this disease. So I was highlighting arthropod-borne plant diseases. So these make up the majority of emerging plant diseases. So you see a change in vector geographic range is one of the reasons why they're emerging, and that's due to globalization and also climate change. We're seeing expanded areas where the vectors are active, and we also see they can be active for a longer period of time. So but prior to coming to North Carolina State, I was at Kansas State where we worked on wheat. And with the warm falls, we saw vectors being active for a longer period of time. And I saw that in my time there. And so this really increases the chances for these insects to transmit viruses to plants. We also have the emergence of new viruses related to these factors. Also, they're complex dynamic systems, because you're not just dealing with a pathogen, you're also dealing with the pest insect. And then we have limited effective control options. And I'll just say, in a lot of areas, especially in the US, we're seeing a lot of the active ingredients that you could use against insects are being phased out. And so now they're trying to find new ways to control the insect vectors. So we're really interested in developing new knowledge of these high priority diseases so we can develop use that basic work to develop control strategies. So this is my case study. So we've been working on tomato spotted wilt virus, and we saw that in Europe and other regions of the world, there were some resistance-breaking strains of the virus emerging. And so I think everybody in this room is more familiar with viruses than they ever wanted to be. We're at the 4-year anniversary around this time of the shutdown from COVID. So you guys are familiar with viruses and spike proteins and mutations and how that affects emergence of these new strains of viruses. So this is exactly what we saw happen with this tomato spotted wilt virus. So since 1998, all the tomato producers in the world have been using the same resistance gene to try and control this virus. And this gene, we call it SW5B, so it's an immune receptor, so it's a protein that recognizes one of the viral proteins. So you can imagine that's an enormous amount of pressure on a single gene. So not unsurprisingly, around 2003 and 2004 in Europe, they started seeing the emergence of what we call resistance-breaking tomato spotted wilt virus. It was able to overcome that resistance gene. And then in California, it was first observed in 2016. So what happens with this virus, so this is, we just have a hypothetical little cartoon here. The blue would be the receptor, and orange is the NSM gene. What happens is a single amino acid change in this gene then enables it to not be recognized, and you have disease happening. So it's really amazing that a single amino acid can confer this ability to overcome resistance. And there are multiple mutations in a single region that enable this to happen. So what happened here in North Carolina is that we had some really great people working in the field. Dr. Reza Shakatstaban and Inga Meadows are both based in the western part of the state, where we grow more of our tomatoes, or a lot of tomatoes. And they were observing virus on these resistant plants. And so they contacted me and my collaborator, Dr. Dorit Rotenberg, and we were able to take that virus and sequence it and determine if it was indeed resistance-breaking virus isolates. And so we worked with a great team of scientists in our group. We have postdocs and graduate students and research technicians that really contributed to this work and going to the field and looking for these diseases. So we sequenced these viruses, and we found 2 mutations in one county of North Carolina, in Rowan County. So I know that, so they have 2 different mutations on 2 different farms. And so this just shows you that. That's really not important for you guys to know. But one thing that we were able to do, and this is as part of our cluster working with Dr. David Rasmussen, who's a virus phylogeneticist, we were able to look at the relationships between the viruses we were finding in North Carolina and the viruses we were finding other places in the world. And so in red here, of course, for North Carolina, we have the viruses that we sequenced here. And then you can see that they fall into a clade here. And then you have different clades from different regions of the world. And then here we've highlighted the mutations that they have. And so one thing you'll see is that the North Carolina isolates have mutations in different spots, and some of these same mutations are the same ones we see in different regions of the world. And so what this phylogenetic analysis enabled us to determine is that it wasn't the same isolate of the virus spreading all over the world. It was that these new mutations were arising independently. So even within the state of North Carolina, within the same county, we had these new virus resistance-breaking isolates emerging. And so that's really important for helping us understand how to control an emerging plant disease, because you're not trying to stop TSWV from coming in. You've got to manage the populations that are developing in your local area. And so, as part of our cluster, we have Dr. King-Shan Wei, who's an expert at developing new technologies for detecting viruses and other pathogens. And so he was able to develop a really fast and simple method for virus detection. One of the key features that I know people in my group really, really appreciate is he developed something called a microneedle patch. So in the past, when we were isolating nucleic acid and trying to do virus diagnostics, you know, everybody remembers the COVID test where they did RT-PCR to detect virus. So that's exactly what we're doing for these plant viruses. So you have to extract the nucleic acid. So he developed this nifty little microneedle patch. And so instead of grinding up really hard to grind up plant tissue and doing all these steps, you're able to just press this little microneedle onto the leaf and then take that and dip it into your buffer, and you can amplify from that. So it's a really easy method and a great technology. So this was really important for detecting emerging diseases. And then in collaboration with Jean and other people in our cluster, we developed a further assay so we could detect that single amino acid mutation that was part of that resistance breaking strain. So we're really developing the tools for people to use in the field to determine if they have this emerging plant disease. And then finally, we use all of this knowledge to then also develop new control strategies. And so working with Dr. Dorit Rotenberg and Reza Shakatestaban, one thing my team did was to take knowledge of these viruses and say, can we develop a durable plant virus resistance gene that will also protect tomato plants from this resistance breaking isolate and others that might come along? So one of the things we did phylogenetic analysis of all tospoviruses sequences that were available. So we went to GenBank, we went to our friends, we said, give us your sequences. We then looked for the most conserved regions, and we developed a transgenic plant that expressed RNA to those viruses, and then screened them against an array of viruses from North Carolina and beyond. But I'm showing here just our testing with the resistance breaking form of TSWV, because that's the one where we really need new control strategies. And so in this graph, I have the percent plants infected with TSWV, and then I have different lines of the plants that we generated, and here are the controls that are susceptible to virus. So you can see we had really high levels of infection in our controls, but very low levels of infection in some of our transgenic plants. So this is a new strategy, and it's gaining in interest. I don't know if you guys have heard about the magnificent purple tomato. That's a transgenic tomato that people can now buy. So I'm optimistic that there is a change in the philosophy with the acceptance of bioengineered crops. And we can talk about that more later at the break, for sure. So just to summarize, global food security is deteriorating, and there's greater need than ever for continued improvements. We think that this knowledge of high priority emerging diseases has led to the identification of this virus in North Carolina and applied strategies to help growers here and globally. And this multidisciplinary approach and the resources that are available here through NCSU really enabled this fast response to this disease. So some of the future directions, our cluster is embedded within we're really integrated with the Global One Health Academy led by Sid. And we're also really hoping that this Predictive Intelligence for Pandemic Prevention Center proposal will be funded. So this would be bringing in a bigger group of people to do genomic surveillance and phylogenetics on several other pathogens, and then developing detection methods and forecasting models. And so this is led by Dr. Chris Jones, and there's several co-PIs from around NC State and from Cornell and University of Florida involved in this project. So we're very optimistic and hopeful that that project will be funded. So with that, on my acknowledgement slide, good timing. This work was partially funded by Plant Aid, and I also want to highlight that today at this Symposium, we also have Michael Bradshaw. He's the newest member of our cluster. So if you see him, say hello. He's going to be working on powdery mildews, another important plant pest. So with that, I think maybe I have time for a few questions.
Any questions?
Thanks, great presentation. So if I'm remembering the timelines correctly, you said that the traditional tomato with the resistance gene had been in use since 1988, but the first resistance breaking wasn't seen until 2003. Or that
Yeah.
seems like a long time. I'm wondering if you can, I'm surprised it took that long. Like, can you explain more about why it may have taken that long? Were the tomatoes not widely used up until that point?
Well,
Or,
you're-
and
so
how that
that's
might.
That's a very good point. I
Yeah,
think there are
how
probably
that might
several-
lean towards resistance to the new transgenic tomatoes you're developing as well.
Right. So one thing is that, so in 1988 was the time when it was first deployed, but tomato growers all over the world grow different varieties of tomatoes because they have different attributes that they need for their growing conditions or for their market. And so it would be a while before, so maybe it was first used in 1988, but then the breeders have to get that SW5 gene into those different varieties. So it wasn't until later that it was like very widely deployed. And I would also say, 1988, we probably weren't doing, in the 2000s, that's when we probably did a lot more virus sequencing. So even if it had been out there, maybe people weren't going to detect it at the time. So I guess it would be very interesting to do a deep dive into some of that older data. But like I said, there's a lot more sequence information out there now. And I'll just contrast the SW5-based resistance from our transgenic. One thing that I forgot to mention was that we are targeting every single virus gene. So that's why we did the phylogenetic analysis, was we wanted to identify the conserved region of every gene. And so what we're thinking is we want to, if it's a conserved region, that means it's probably highly important in the virus infection cycle. So if it made a mutation in one of those regions, hopefully that virus will then no longer be alive and able to replicate. And so that's why we did target 5 different genes. Because this virus is able to generate lots of mutations. One thing I didn't talk about, it has 3 genome segments, and so it's able to swap when it has mixed infection. So it changes its genome really, really rapidly. So we're throwing the kitchen sink at it. Okay.
Well, that was a scary but hopeful presentation. Our third speaker today, and I hope you will see the theme of this morning's highlighted speaker, is Dr. Francis de los Reyes. He has so many titles, I'm not going to even start. The only thing is he is a distinguished professor, and he is the director of WASH, the WASH Center here at NC State, Water, Sanitation, and Hygiene. Dr. de los Reyes, he is collaborates with companies, universities, and non-governmental entities in Europe, Asia, and sub-Saharan Africa. And it doesn't need a lot of explanations to explain how water supply and water and sanitation fits into not just phosphorus recycling, but also food security and nutrition. So thank you, Francis.
Okay, thank you, Heike. Good morning. About 7 years ago, I was with a group of researchers in India, and we watched a private company. It's a pit emptying or septic tank emptying company, empty a septic tank from a village, and then we followed them to this farm where they basically dumped the untreated septage, the fecal sludge, into this farm that was used for growing human crops. So I think it's quite obvious that there is this relationship between food security and safety and water. So we know that water access, stability of that access, supply of water, and the quality of that water affects how you produce food, and not just in terms of agriculture, but also in terms of food preparation and hygiene, and obviously health. So this is a diagram from a paper that I thought summarized some of those connections. But there's also a very direct link in water and sanitation and health. And this is the classic 5F diagram from the 1950s. 5F because it's feces, fields, fingers, food, and so on. And the barriers there are actually the barriers that proper water and sanitation infrastructure should provide. So if you have clean water for hygiene, for washing, and so on. So this obviously is not happening in a lot of countries. So a lot of our research is global, as was mentioned by Heike. And about 800 million people around the world still don't have access to clean drinking water. And so this is surface water collection, which is the dirtiest form of water for households. And about 3.6 billion people around the world, almost 50% of the world's population still don't have access to what we call safely managed sanitation. In other words, they have a facility at their house, and then their waste is properly treated and separated. And so here are pictures. On the left is a picture of a manual pit emptier. That still happens today. Talk about 21st century global health challenges. In the middle is, once they collect that septage or that fecal sludge, usually they just dump it into the nearest body of water. The left is a picture from Burkina Faso. The middle one is from Kenya. And then the last one is a picture I took in Zambia, which is just an elevated pit latrine. So this obviously relates to diseases and health implications. In about 829,000 deaths, you can do the math, that's about 2,000 something deaths per day, which is about 60% of the total diarrheal deaths. And we have evidence that if you actually invest and put proper interventions that can actually prevent a lot of these deaths. For example, if you have access to safe drinking water and adequate sanitation and hygiene, you can reduce the global disease burden by 10%, according to WHO. And then in Southeast Asia, East Asia, and Oceania, you can decrease deaths, diarrheal deaths by 15%, and then globally by 10%. So we know that this works. We can think of this in terms of the SDGs, the Sustainable Development Goals. So here's an interpretation where SDG 6, which is safe drinking water and sanitation, is at the middle. And you can see that in the immediate vicinity, you have SDG 2 and SDG 3. So just above it, zero hunger, and also good health and well-being. And you can see how these are all interconnected. So I lead the Global WASH faculty cluster. And what we do is we analyze these pathways. How does water and sanitation actually impact the pathways of pathogens through food, through water, through exposures? And then how do we actually assess some of these interventions? There's really a huge area of research in terms of figuring out the actual indicators that actually work and tell us the effectiveness of interventions. And then beyond that, we research the technologies that can be the basis for these effective interventions in the context of emerging threats like AMR, antimicrobial resistance, and urbanization and climate change. And then finally, it's not just all technology, but looking at incentives and the economics, the enabling environment around these interventions to make them work. So that's what the Global WASH faculty cluster does. But this is an RLA meeting, so I wanted to focus a little bit on research. And I wanted to talk a little bit about one of my research areas, which is the sanitation chain. So we call this the sanitation service chain, the sanitation value chain. It starts on the left with the user interface, which is the toilet. And then that waste has to be stored, collected, transported, treated, and then hopefully eventually reused. Now, we don't see this. This is all invisible to us because once we flush our toilet, all those pipes are actually the collection and transport, right? And then the treatment, I don't know how many of you know where the wastewater treatment plant is that treats your waste, but it's all hidden, right? We don't see that infrastructure. But in low and medium income countries, this is quite visible. We have pit latrines, we have septic tanks, and then the only way that you can collect is with vacuum trucks that drive through the city to hopefully a proper treatment facility, or if not, the nearest river or body of water. So I wanted to focus on some example research that we do as examples, and then go back to the major challenges. So on the front end, we can do things like CFD, computational fluid dynamics, and say, how can we design a toilet that can flush with less than a liter of water? And that requires actually studying the fluid mechanics and new designs of toilets. If it's a flushing culture, so not every culture is a flushing culture. What are the microbial processes occurring in pit latrines? And so these are just basically holes in the ground. You have a slab, and then you have basically your infrastructure, your toilet above it. And this is very common. The question is, what really happens in these things? And so here, we put on our microbial ecology and biotechnology hat and say, hey, we do things like sequencing of the 16S rRNA gene, the gene sequencing. We can figure out what's going on here. And so with our partners in Malawi, we sampled pit latrines, sampling them at different depths, and then doing also questionnaires and trying to correlate these to different factors in the field. So here's a summary, which is basically a way of envisioning the microbial communities. And this graph is essentially showing you the different samples, the different microbial communities. And the closer they are to each other, the more related they are. And essentially, the conclusion here is that if you just look at the pathogens, then actually sampling from the surface or the middle or the bottom of the pit makes a difference. There are differences in the pathogen community composition. We can also take a look at the actual functions. And so we can say what's happening at the different depths in terms of protein metabolism, methanogenesis or methane formation, and short-chain fatty acid metabolism. So this is something that we can see and figure out, okay, what's happening at the different depths, and then from that, provide a picture of the transformation pathways within the pit. Here's one that I wanted to show because this has implications in terms of climate change. A lot of these pit latrines actually produce methane, just like your septic tanks. I'm on a septic tank, and my septic tank I know produces methane. It's something that in the IPCC calculations we're trying to figure out a better number for estimating those greenhouse gases. But we found out that in the pit latrines that the methanogenesis pathway is actually quite specific to that particular pit latrine. We thought that there was going to be a combination, but mostly it's either hydrogenotrophic or acetoclastic, and we were quite surprised by that result. So just an example of how we are using molecular techniques and advanced gene sequencing techniques in this context. Another technology is looking at pit latrines. I told you about manual pit emptying, but how can we make this safer, more dignified, more efficient, and more profitable for those services, for these companies? So this is a slide that summarizes about 10 years of research, going from the field to the lab, making mistakes, licking our wounds, and then going back to the lab. And in the end, we have a couple of technologies, the flex curvator and the excluder, that a few years ago won the Patents for Humanity, US Patents for Humanity Award, and then also the RELX Grand Challenge Award. So this is technology that's ready to use. We can talk about commercialization and what's going on with that. That's a whole field that I can describe it, and all the challenges. Now, I talked about work globally, but also in the US, we have lots of challenges. About 2.2 million Americans don't have access to basic water and sanitation. And then we have these millions of Americans who have these, what we call straight pipe solutions, where you basically flush the toilet, it goes back into the backyard, untreated. And you've heard probably of Lowndes County, Alabama, which is a famous situation in the US. But those communities are all over. And we have millions of Americans who basically have this situation. And most of these small systems are contaminated. We have the data for that. And then this NGO, Dig Deep, did a calculation and said that it costs the US economy about $8.6 billion every year. And of course, you know about our failing infrastructure, estimates from $600 billion to $1 trillion over the next 2 decades to catch up. So how do we rethink and transform this infrastructure? The first is that we need to develop technologies that are different from what the centralized system that we have right now. Not everybody can have the flush toilet with the sewage pipes going to these large centralized treatment plants. So we need to think about technologies at different scales. And so here we're showing technologies at small community scale, at household cluster scale, maybe a dozen houses that share the same treatment, or the most decentralized scale, which is at your house, right? How do you reuse your yellow water, your gray water inside your house? The second insight doing this work is that we need to work with communities. A lot of these technical solutions don't work because we don't actually go out and co-design and really engage the communities that we're working in. Context matters. There's no one size fits all, and the solution for one community is going to be different from the solution for another community. And finally, how do we think about really sustainability and resilience in the face of climate change? So what this graph is showing, or this slide is showing, is how we're starting to think about scale and convergence and communities. I'm really very pleased to have a few papers now out with social scientists. You know, I've always dreamt of having papers with social scientists and learning from them, and it's happening, and really excited about that. So that's what we're learning. As an engineer, I need to actually retransform and think about myself in a different way. And I can talk to you a little bit about that too, how I think engineering should actually become more data-enabled and community-based. So we are waiting, a lot of you probably know that we are waiting for funding, word of funding or not funding, an ERC, an Engineering Research Center. We've done the site visit, we've done the reverse site visit, it's really just waiting time right now. But we call that ERC Clear Wash, community-based learning and engagement to achieve resilient wash. And it's a lot of text, but basically the vision is sustainable, resilient, and inclusive infrastructure for everybody, with a focus on underserved communities. The second is this concept of context-specific solutions at different scales. And to do that, and if we do that, then we can achieve this goal of reducing our water use and also reducing our energy use and greenhouse gas emissions. I wanted to leave you one idea from that proposal, which is this House of the Future of Water. The idea is that in the future, within our houses, we will actually treat our water on the spot, and we will not be using clean drinking water for flushing our toilets. We don't need to. We will have the technology at our house that will separate our yellow water, our gray water, and treat our water so that it's mostly reused within the house. This is something we want to build, a real house with real people, and show the technologies really work. Hasn't been done in the US before at this scale. There is an office building in Switzerland that kind of does this. But this is the prototype of what we see as the future. And so hopefully we'll get an email from NSF soon, or a phone call or something. But if not, life goes on, and we'll keep doing what we're doing. Thank you.
We'll probably skip questions because we're going to move to a break. So if you have questions, I think, I do still want to give the group a 10-minute break. And then I'm going to ask us to be back in our seats at 11:10. If you're a faculty member that's part of our first round of lightning rounds, please meet Sophia kind of around this table that has a reserve sign closer to the break station. So if you're part of the morning session for lightning rounds, go ahead and find her after you do a bio break or get another cup of coffee. But let's say back in seats by 11:10, and we'll get started with the next section of the agenda. But try to make a couple connections maybe in this first break, and we're going to have lunch, and we'll have a second break this afternoon. But 11:10, back to your seats, and we'll continue our program. Thank you. All right. I know 10 minutes isn't enough time to connect, and I promise we have lunch and all coming up later, but I am going to ask that you all, we sit back to our seats. If you haven't checked in with Sophia yet and you're part of our round one for faculty lightning rounds, please do so right now at that reserved table. So while that is getting organized for our first couple of presenters, we're going to turn our attention in our program now to what we're calling a rapid fire presentation style of some amazing research that's happening here at NC State. This next section of agenda, we have faculty, we're giving them a chance to showcase their research and provide a platform for our NC State community to discover research across campus. Our presenters are going to have only 2 minutes to present their research or scholarship as relevant to any aspect of 21st century threats to health. So we have 8 individuals here in our first round, and they're starting to line up. We're going to allow each of the presenters to introduce themselves. And my only item I have, and Sophia might have said this for you all as the presenters, once you're finished with your 2 minutes, please press over or advance one more slide so we see the next person's name. Okay, I'm going to ask Rosemary to come up to the stage, and you'll be our first presenter.
All right, thank you so much, everybody. My pleasure to be here today. My name is Rosemary Bayless. I have a lab at the College of Veterinary Medicine, where I'm also a veterinarian clinician scientist. So today I'm excited to tell you about some of our work investigating new therapies for asthma. So asthma is a disease that affects, this is just the United States, so 25 million people. Multiply that times 10 for the number of people around the world with asthma. And in patients with asthma, as you see on the right side of the lung, they have inflammation and airway constriction that cause the clinical signs that we recognize of shortness of breath, coughing, which can progress to needing emergency treatment. And what my lab focuses on are neutrophil-mediated diseases. So neutrophils play a key role in the pathophysiology and driving the clinical signs of 6 million of these patients with asthma. And what I'd like to highlight is patients with neutrophilic asthma tend to have more severe clinical signs and therefore really are an important but underserved population. The risk factors for neutrophilic asthma include environmental exposures such as air pollution and agricultural organic dust, also some more personal factors such as cigarette smoking and obesity. And since the theme of today is increasing threats in the 21st century, we all know that a lot of these, we talked about food production here. We know about the increasing obesity, and so the burden of neutrophilic asthma is likely to grow in the 21st century. Now, in general, asthma is treated with inhaled steroids. There are some newer monoclonal antibodies that are injected as therapies, but one gap and one challenge in clinical need is the fact that neutrophilic asthma these patients tend to not respond to these traditional therapies, which really highlights the need for novel therapies. And so my lab, we use our background in veterinary medicine and comparative health to, number one, identify new drug candidates via in vitro studies. We move them into a mouse model to test them and verify their efficacy. And then we actually have a naturally occurring model of asthma, neutrophilic asthma, in our horse populations. And then I'm also connected with some folks at UNC. And so I have some human physician clinician scientists. And that's my happy place in the middle of basic science, human health, and animal health. Thank you very much.
So my name is Michael Bradshaw, and I study emerging fungal plant pathogens. Anna already gave me a great introduction, so thank you. So first I want to emphasize that how understudied fungi are. Around 150,000 fungal species have been described of the estimated 3 million located throughout the world. One very famous mycologist, Amy Rossman, once noted that for some types of organisms, such as microfungi, New York State's forests are almost as unexplored as the tropical forests. And I want to emphasize this plant on the bottom right corner. This is a lupinus plant covered with powdery mildew, which is the study organism that I study. And you can see this plant is completely covered with powdery mildew. This was found right in front of the biology department, and this was an undescribed species. So I'll take this quote a step further and say that North Carolina State University campus is almost as unexplored as the tropical forest. I usually I use herbarium specimens to study. They can answer all sorts of interesting questions. It's a great underutilized resource. Some herbarium specimens are over 2,500 years old. For example, there's a specimen at a herbarium that is a lichen that was wrapped around a mummy. And just imagine the stories that genome could tell you. So research on herbarium specimens has yielded key insights in determining the genetic structure and evolutionary history of plant pathogens. Here's a big leaf maple covered with powdery mildew from the Pacific Northwest. This is a really important ecological plant. And sequencing herbarium specimens of this plant showed me when this invasive powdery mildew was introduced into the United States, and two, when the epidemic started. Here's a new system that I'm starting to study. This is powdery mildew on blueberries. This disease is now spreading throughout the world and impacting the blueberry industry. We estimate that this could cost around $500 million per year to control. Here you can see what each one of those little black dots looks like. Each black dot holds around 100 spores. And then I just want to briefly give a shout out to Dr. Jean Restaino. She was a pioneer in this type of research where we sequence herbarium specimens. And her research found that the genotype of Phytophthora infestans that caused the Irish potato famine is genetically distinct from the genotypes present now. All right, thank you.
Hello, I'm Ralph Dewey from the Department of Crop and Soil Sciences. And let's see. Yes, first I want to start by what has become a favorite quote by plant breeders worldwide, and it's found from the classic novel Gulliver's Travels by Jonathan Swift. It goes, "Whoever could make 2 ears of corn or 2 blades of grass to grow upon a spot where only one grew before would deserve better of mankind and do more essential service to his country than the whole race of politicians put together." So I think you'd find that there are few in this room who would argue that maximizing the yield potential of crop species, which is another way of saying growing the most amount of food on the least amount of land, is critical to global food security. So from a plant genetics point of view, what has been the most proven, time-tested ways of accomplishing this has been by exploiting a phenomenon referred to as heterosis or hybrid vigor. Now, a good example of this is shown in the crop species of maize, where you can take 2 inbred parents of different heterotic groups. When you cross them, you can produce a hybrid that is far more vigorous and higher yielding than either of the parents alone. Now, making these crosses and producing the type of seed at a commercial scale is non-trivial, and it is most typically done through a trait referred to as cytoplasmic male sterility, or CMS, which is defined as the maternally inherited inability to produce pollen. Now, CMS is caused by mutations on genes in the mitochondrial genome, which explains their maternal inheritance, as mitochondria are strictly inherited to the maternal parent. So where available, in crop species where available, the CMS systems are widely used to maximize the production of high-yielding hybrid seed. Unfortunately, CMS systems, naturally occurring ones that are efficacious, aren't available in many crop species. So what my lab has done is, using tobacco as a model system, we have developed a CMS-based system of hybrid seed production. Briefly shown here, wild-type tobacco anthers produce abundant pollen. Our mitochondrial genome-edited anthers from the tobacco produce none. Now, the CMS line is just one of the essential elements of hybrid seed production. The others, you have to have what's called a restore line and a maintainer line. We produced the entire 3-line system in our tobacco model. As a first step in demonstrating that this can be applied to other non-model species, we have introduced our system in the tomato, where we show here wild-type stained tomato anthers produce a lot of little pollen grains. whereas our mitochondrial mutant anthers produce none. In theory, this system should be able to be applied to virtually any crop species to help facilitate production of hybrid seed. And with that, I thank you for the 2 minutes of your time.
Hey there, and thank you. My name's Megan Manfra. I'm a professor in the College of Education where I specialize in digital integration in both in the classroom and when we work with students. So, open question for us, is social media a 21st century threat to health? I think that there's some evidence to suggest that perhaps, but the extent to which it is a threat has been sort of under-researched and really fallen into the realm of politics and policymaking. You may have heard that there's a threat to shut down TikTok that's sort of making its way through the US Congress. In response to this, thousands of middle schoolers contacted their congressional offices saying that they did not want TikTok to be banned, which and to my mind might be a nice example of some civic engagement here. So maybe there is something to be said for the opportunity to have kids engage in policy making. Closer to home, school systems are threatening to sue social media platforms. You've seen this across the country. Organizations like the National Institute for Health, the World Health Organization, the CDC, or Surgeon General in the US, has said that we really don't fully understand the risk of social media has on our students. So what are we going to do about it? Part of my research takes a two-pronged approach. The first is to work with students and their caregivers to really think reflectively about the way in which they engage with social media. We know that adolescents are particularly vulnerable to the social effects of social media because of the way their brains are developing. But at the same time, we can also work with educators, both at the K-12 level and beyond, to think about really kind of teaching systematically media literacy and the types of skills students need to navigate this supposed threat. So, thank you.
Good morning, I'm Stacy Wood, and I'd like you to consider whether a professor of marketing might not be a great addition to your projects and papers. I would love to be the co-investigator or co-author with my STEM colleagues. I dream of it. I study the psychology of innovation. So how do you get people to make those changes, choices, and decisions that get people to adopt the incredibly genius things that all of you are coming up with? Now, I started studying this in the mid-1990s in the brand new world of e-commerce. I wrote one of the first papers on why people might be in any way motivated to either buy goods online or sell goods online. And I feel like it's certainly rolled from there. So I study innovation primarily in technology and in global trends, everything from cultural trends to sport trends to sustainability. And more recently, starting about 2015, I studied this in healthcare. Right now, I'm doing a lot of work on the intersection of technology and healthcare, like how AI might have the potential to try to help with medical disenfranchisement in the US and in other countries. So what have I learned? Basically, getting people to do new things is really hard. It is not intuitive. It is not, as my engineering father said, just a matter of doing the right thing. Good luck with that. Now, this all came to an amazing kind of moment of impact for me during the pandemic when some colleagues and I wrote a couple of papers for medical journals on how to use marketing theory to promote pandemic-relevant behaviors like social distancing, masking, and vaccine promotion. And for a scientist who was not used to people reading her papers, this was quite a boom time. But it goes to show that I learned that I need to be a louder advocate for marketing. While it seems like a non-science to many in the academy, it is actually critical for that transition from bench science to established practice in the real world. So think of marketing next time you're putting together a grant. Thank you.
Okay, good morning, everyone. And first of all, I'd like to thank the organizers for giving me an opportunity to talk this morning. Okay, my name is Kerim Eroglu, and I'm an assistant professor of biochemistry at Plans for Human Health Institute in Kannapolis, North Carolina. So today I'll be talking about microbial carotenoid metabolism in obesity. There it is. So carotenoids are naturally occurring pigments primarily synthesized by plants and microbes, and they have multiple important roles with respect to human health. They can display some anti-inflammatory and antioxidant properties. As you know, obesity is a state of chronic inflammation characterized by low-grade inflammation, and we tested whether a diet rich in carotenoids can display anti-obesity effects. And then to our surprise, in a mice model of obesity, we found out that a diet rich in orange carrots displayed these anti-obesity effects. They lowered the body weight gain while eating actually more, again, to our surprise. So our motivation is whether the biological activities of carotenoids in the large intestine can be mediated through gut microbiome. So with respect to metabolic health, it is well appreciated in terms of their role in the host organism, but how carotenoids can mediate their biological activities through gut microbiome is relatively underappreciated, and that's what we try to achieve. So we came up with these big, beautiful biology questions. I really like this, using this phrase on and all the time. So one question was how do carotenoids modulate the gut microbiota in preclinical model of obesity, and whether microbial-derived carotenoids can improve the human health. So to address that, we recently wrote an article summarizing both opportunities and challenges, and it was featured in Advances in Nutrition just a year ago. So my lab is trying to understand the dynamic interplay between carotenoids, gut microbiota, and obesity using a systems biology approach and a genetics approach. Metagenomics, transcriptomics, and proteomics is just a few of them. So our project is recently funded by the USDA and NSF, and hopefully it will be funded by the NIH in the coming months too. Thank you so much for your attention.
Hello, everybody. Robert Hayes with Nuclear Engineering. Usually I think that my research is the coolest out there. There's a lot of really cool stuff going on here, so we'll see how mine stacks up. So to start off with, I do radiation safety, radiological protection. So let's do a quick little overview of that if we can, and go over that. So 1 millirem, that's about what you all get every day just from natural background radiation. Kind of contextualize that. That's what this little planet Earth is over here, giving 1 millirem per day. 5 millirem, that's if you do a coast-to-coast flight, just from space radiation. It's also the annual drinking water limit from the FDA, or sorry, from the EPA. We're doing logarithmic scales here to kind of put in perspective the degree with which people are generally not able to assess radiological risk when it comes to nuclear technology. 40 millirem, that's about the average dose that you get every year from internal potassium. Potassium is a primordial radionuclide, and it gives you about that dose every year if you're healthy. 100 millirem, that's about the dose you get for a pelvic X-ray. It's also the annual limit to a member of the public for a nuclear facility, such as NC State, or a nuclear power plant, or any other nuclear facility. 320 millirem, that's about what you get every year. So 1/3 of that is what's legal to take place, maximum dose to a member of the public. 320 milligram, that's what you get from all sources combined from nature alone. 1,000 milligram, that's a legal dose to a radiation worker. 5,000 milligram is a legal dose to a radiation worker. 1,000 milligram, 1 rem, that's the threshold at which the EPA could issue evacuation recommendation for a nuclear event. Put that in perspective. You can actually get upwards of 10,000, oh, sorry. Yeah. So I've already gone over? Okay, well, I'll speed this up. The point is that at 1,000 milligram, that's where evacuation can take place. But 5,000 milligram is a legal dose to a radiation worker. It's considered sufficiently small. 10,000 milligram, now we've just hit a new threshold. That's where cancer probability has first been discovered. You have to get 10 rem just to have a statistically significant increase in cancer probability. All these doses back here are below any measurable medical effects, kind of putting that in perspective. It's not up until you get to 100,000 milligram where you start to get acute radiation syndrome, and it's not until you get to 1,000,000 milligram where you can actually start talking about lethality. And so these values up here are regulatory limits. These values down here are where you can actually get measurable medical effects. And this is logarithmic. Now, where that comes in context is where you compare that to things that scare people. 3 Mile Island, for example, the average dose to the member of the public was 1 milligram. That was the smallest dose on that scale. It terrifies people. If you go up to Fukushima, much higher. There was a potential to get upwards of 1,000 milligram, 2,000 milligram. Again, below measurable medical effects, but enough to terrify people because of the evacuation. In fact, if you go to what the United Nations said or the World Health Organization, all the papers, they all agree that all of the radioactivity from the initial Fukushima was insufficient to produce any measurable medical effects in the Japanese public. And now people are terrified of the water releasing tritium, which is a naturally occurring radionuclide, at levels that are insignificant compared to natural levels, and yet it terrifies people. So a lot of my research is in this, and I guess I'm way over, so I'll just go ahead and end it there.
Okay. This was very rapid fire, but I thought it is designed so you can see what breadth of research from and marketing and psychology, all the other aspects that are so important for bringing our ideas, our natural science ideas and developments into actual use. The next thing is our first panel, which will be chaired by Sue Fenton. Sue is the director of the Center for Human Health and the Environment, CHHE, here at NC State, and you're going to introduce your panel yourself. Astrid and Sid, if you can come up.
Thank you. So, thank you to the organizers, Heike. Thank you so much for the invitation. And I just wanted to quickly give you a little bit of an intro to the Center for Human Health and the Environment. If you don't know me, I've only been here since October, and I'd love to meet you afterward. But the coordinated mission of our center is really to understand how human health at both the individual and population level are impacted by environmental chemicals. And then use this knowledge to either prevent or reduce the adverse impacts of the environment on human health through integrative communication and research strategies. So I was paying attention on that marketing talk. Our center is not just NC State. We are also NC Central. We are also ECU. We bring together a really diverse group of people. And we represent, but the NC State faculty represents 7 of the 12 colleges and 17 of 68 departments. So we are really diverse, and we were first funded in 2015. Now, here's a little glimpse at those colleges that are represented so that you can kind of see where we're representing. In CHHE, there's administrative cores, there's a facilities cores, there's communication, and translational research support. So to get us access to biobank samples, things like that. The programs, though, are really where everything starts coming together, where you have career development, funds that can support junior investigators, and then pilot projects that are $25,000 projects open to anyone at the university that can spur new areas of environmental health science research. But really, it's how this all comes together and how we communicate our message that is the best part. And that is done on these research interest groups, these thematic research areas in behavior and neuroscience, water and emerging contaminants. We've heard a lot about that today. Climate change and airborne contaminants, genes, environment and precision health, and environmental health disparities. We have about 25 people in each of these research areas that are kind of focused in those areas, and their work leads the field. So today, we're going to hear from myself just for a few minutes on endocrine disruptors and how consumer goods are filled with them and how climate change can change our exposure to them. Then Astrid's going to go next, and I'm sorry I have these out of order, but she is the director of NC State's Ocean and Human Health Center, which was recently funded. Couldn't be more happy. This is the second center kind of funded out of some pilot projects that were in our CHHE. And then Sid Thackers, who is the executive director of the Global One Health Academy, is going to follow with some discussion of antimicrobial resistance and infectious disease. And I just want to give a shout out to some of the talks that are coming later from some of the other CHHE members like Andy Binder, Kara Grieger, and Aaron Hip. And I'm sorry if I missed anybody, those are the ones I saw on the agenda. So endocrine disruptors are exogenous chemicals or mixtures of chemicals in most cases that interfere with hormone action. And there are 85,000 chemicals at least registered in the marketplace under TSCA for consumer use. However, only 10,000 or so of those have been tested in any kind of assay, mostly in high throughput screening assays, which are kind of an artificial setup in a lab. And of those, about 100 of those, or hundreds of those, maybe a couple hundred, are endocrine disruptors, known endocrine disruptors. They are known to either in an animal, a human, or in a dish, alter endocrine action. Some of the endocrine disruptor sources are shown here. I would say bisphenol A is not just bisphenol A, it's all the phenols. There are a lot of them. There's a large family. Dioxins and PCBs we've heard a lot about lately because of Poe Hall. Flame retardants are things in your furniture, carpeting, meant to, and actually all your electronics, meant to reduce friction and flame. Phthalates are things in anything that has a smell. They're in plastics. Pesticides you know a lot about, I'm sure. Phytoestrogens are in soy. And PFAS are something I'm going to talk a little bit about more, per and polyfluoroalkyl substances. So the big deal about endocrine disruptors are that these things are in almost all consumer goods. They can change the levels of hormones. They can interrupt how your body is supposed to act to normal hormones in your body, or they can actually change the way your body reacts completely to your normal hormones, or they can interrupt the ability for them to act normally. So it's kind of a big deal. They're in everything that is on the market. And PFAS in particular are one that I work on quite a bit. The spots that you see here are PFAS contamination sites just in the US alone. They're known globally to be affected, and every single one of you in this room has this in your blood. You're born with it, unfortunately, at this point. Each child is born with PFAS in their blood. So this is a big problem. There are at least 12,000 PFAS, so it is the largest group of endocrine disruptors that I'm going to talk about. And military firefighting foam is one way that on Air Force bases and any place that has planes, because it puts out jet fuel fires, is contaminated with. And so also people who make PFAS have also contaminated waterways across the globe. So I just want to say one thing about climate change, in that it is going to change how we're exposed to so many things. I think we need to think about climate change in big ways. So it's going to, like extreme storms can move EDCs to areas where they're not known to be located previously. It can stir sediment, which releases things like PFAS that would normally have stayed there. It can increase fire events, and that would increase firefighting products that we use to put out the fires, getting into our environment, and the things in our home that contain all these EDCs are then released into the environment. Flooded intense rain can move contaminated waste into community water supplies. That has been shown numerous times, and I think all of us remember the pictures of pigs in the water in the Eastern Carolina when there was a hurricane, and all the pig waste that then was posited onto crop and animal food growing areas. Drought and global temperatures also can be a problem because there's going to change pesticide use patterns. And some of the infectious diseases that we heard about this morning, it'll concentrate waterborne endocrine disruptors such as hormones that, like for women who take contraceptives, a lot of the hormones go into the urine and they're then into the wastewater, and those are then concentrated and can affect amphibious and fish and other wildlife. And then also it can also directly affect human health by changing fertility patterns and excessive heat. And also chronic kidney disease of unknown origin is thought to be closely linked with excessive heat and dehydration in worker populations. So last slide from me. PFAS contamination in North Carolina is no joke. GenX is something, or HFPODA is what the chemical name is for it, but GenX is the regrettable replacement for one of the PFAS that were made in Fayetteville. So it began entering the Cape Fear water system in the 1980s. Detlef Knappe here at NC State and many colleagues who I've been close friends with for a long time discovered this in probably about 2013. But it wasn't until about 2015 that it caught fire and like everybody started paying attention. The government of North Carolina, actually took away the permit for Chemours to put this into the water in 2017. But we now know that GenX causes the same health effects as PFOA, the thing it replaced. In fact, it causes the same health effects at lower doses than PFOA did, and it doesn't have to stay in your body to cause those effects if you were exposed as an infant or a fetus. And these are animal studies where we did have causative relationships. Human studies are not many on this right now, but as you can tell from this long history of exposure here, there's a very good possibility that we'll be seeing health effects of GenX in years to come. So with that, I'd like to introduce to you Astrid Stechter.
Thankfully, due to Jacob's talk earlier, if you were here, some of the introduction about harmful algal blooms, which we're studying, is going to be very quick. My name is Astrid Schnetzer. I'm in the Department of Marine, Earth, and Atmospheric Sciences. And we study algal blooms. And algal blooms do really well. I'm good at growing algae, not so much with my plants at home, but you know what they need: water, nutrients, and sunlight. And we're providing plenty of that in estuarine and coastal systems. The sources are pretty diverse, including agricultural runoff and whatnot. And then especially in embayments where you don't have a lot of flushing of the system, you have these algal biomass blooms building up. Sometimes when they start to degrade, you have fish kills because the oxygen is being sucked out by decomposition processes. And these are some of the issues that you probably read and hear about within our areas and within the Albemarle Palmico Sound. So eutrophication, over doing it with nitrogen and phosphate, but then there's also an increasing number of studies that show that climate change plays a role, not just in regard to how fast and how severely these nutrients are flushing into systems with severe storms and other things, but also there's some laboratory studies that show that those algal strains that are the toxic ones do better in increased temperatures. There is marine algal blooms, and then there's freshwater algal blooms. The ones that I want to focus on, although my lab studies both, is the ones that make these green lime colors. How many of you have seen that? Show of hands. Stay away from it. Don't let your pets go in there. These can produce toxins, right? You do not want to be close to those. This is 2019 in the Chowan River, which was a really active bloom year. Many of the toxin producers, like the Florida tide and marine producers, are actually microeukaryotes. When you have the freshwater, that's blue-green algae or prokaryotes. There's many taxa. Several of the taxa produce multiple toxins, so I'm not going to go into detail, but it's a challenge and an interesting thing to study. Clearly, some of the issues are sort of like non-brainers. The fish kills I already mentioned. There's studies like in Lake Matamosquite where the submerged aquatic vegetation has been completely smothered out by those blooms in the water column. There's millions, tens of millions of dollars every year that socioeconomic losses cause. There's a halo effect. Once you know that blooms are in that area, you stop eating from those restaurants, you stop going into those places. And then, obviously, when toxins are in the play, we're really concerned. What are some of the concerns? There's several exposure pathways that these algal toxins can take. In their cellular form, which means they're still in the cells, you can get them accumulated over the food web. Each and every single animal portrayed on the slide has been implicated with having these toxins or some cyanotoxins in their systems. Obviously, somewhere in that food web, I should have put a human there, is us, so we're concerned. Once the cellular toxins are released and leak from the cells, we have them in the dissolved phase, then dermal exposure through recreational use is an issue, and also water treatment resource reservoirs are a concern. If your water treatment plant that delivers to you has all the bells and whistles, it is taken out pretty efficiently, but not everybody has that luxury. So food web transfer is an issue, drinking water, and then there's also an increasing number of studies that shows that these toxins can be aerosolized, and our lungs are one of the best filters you will find. Going where I want to go, which is our Ocean and Human Health Center, there's also increasing evidence that you have these freshwater blooms in the low salinity or freshwater areas, and then they get flushed all through the estuary. So we have several preliminary studies where we show these cyanotoxins in marine sounds in oysters and other seafood, so we have this issue all over North Carolina. Challenges, and I also want to say that all the research that had built up to us being able to submit for an Ocean and Human Health Center came from Sea Grant, the Water Resources Resources Institute, and then as Sue mentioned, the Center of Human and Health, wow, I'm really bad with acronyms, CHHE, which I'm a member of, and which really brought us together with biomedical epidemiologists and other experts. My downside was always that I would look at the algal ecology, sometimes look in the animals, and then put those two sentences in the discussion going like, this could have impacts on humans, right? And now I have this wonderful opportunity to work with a team who can actually follow that through and look at those effects, which is one of the wonderful things about interdisciplinary research. So here's some of those things that make it hard. These blooms come and go really quickly. They can happen within days to weeks. They're patchy. This is, hopefully the video is gonna work right here. This is a collaborator from the Division of Marine Fisheries. You see the Chowan River right here. This is a bloom. So if you sample at the wrong location, you won't know that there's a bloom going on, right? So this is how patchy it can get. So looking at them and studying them can get a little challenging. Not all species are toxic, so if you see a lot of bloom biomass, that doesn't necessarily mean there's toxin associated with it, which makes it hard to use biomass as a reliable indicator. Not all toxic species continually produce toxin, more than one toxin per species. Presence of multiple toxins, most of the surveys that we're doing, we find at least 4 out of the 5 that we're looking for. And then it goes into toxin mixtures, guidelines, and so forth. So it gets complicated pretty quickly, and what's the fate of the toxin? Which brings me to the most exciting slide in this intro, is that we used what we had studied to submit for an Ocean and Human Health Center that was just recently funded and has collaborators from 5 different colleges in North Carolina at NC State. You see the graphic right here that encompasses what the North Carolina Center for Coastal Algae, People and Environment, NCCCAPE, is focusing on, and we're looking at emerging and future threats to human health from algal toxins within North Carolina. This is one of 6 U.S. centers. We're in good company with Scripps and Woods Hole Oceanographic Institution, and we're really, really excited to get going. There's 3 research projects. Number 1, the bluish one, is about field studies and looking at the toxins both in water, mainly in the Pamlico Sound, all the way up the Pamlico River. We're looking at blue crab and oyster contamination and look at bioaccumulation. This is with Tal Ben-Horen, who is from the veterinary school, myself and Ryan Pearl, who's also in MEAS. My co-director, Scott Belcher, is here, and if you have any questions about biomedical stuff, I will immediately point you in that direction. Who's working with Catherine Hoyo using mouse models and organoids. I've learned a lot during the submission that was completely over my head to begin with. Then we have the third component here, modeling and climate change. Dan Oberner, Natalie Nelson, and Casey Dietrich, who will feed the information we get from the system, from the water, from the plankton, from the animals, and put them into models and put them in context of climate change. And this is a really exciting part of it because it will also allow to look at risk maps and tie into communities and locations where this is more of an issue than in other regions. And then at the very heart of all of this is Bethany Cutts, who leads the Data Justice Community Engagement Core, which will connect with the communities most affected in the area, and it all involves around data justice, which is a new principle that I also learned about. So again, for me, this is a very humbling and exciting opportunity because I'm learning so much from this team that I'm really excited to work with. And again, huge kudos to people at NCC Grant and CHHE who take a risk and actually allow you to venture into directions that you didn't really have a lot to go by, but it can pay off. And this is NSF and NIH, a co-funded project over 5 years, so we will be able to produce some really interesting results. Thank you.
Thank you. Hi, I'm Sid Thakur. I'm the Executive Director of the Global One Health Academy, professor here at the College of Veterinary Medicine, and I want to tell you a little bit more about what we have been doing at this academy since our inception a little over a year ago. It's one of the newest of the 3 academies based in the Provost office, the key idea over here is that we are not just focused on human health, but we are focused on human health, animal health, plant health, and our environment altogether, and to really connect it with our stakeholders, with our folks at NC State and beyond. NC State, many of you may or may not know, is a World Health Organization collaborating center in One Health and antimicrobial resistance. This is one of the 3 collaborating centers around the world, the only one in the US that has this designation. We have had this title for around 4 years, and we are in the process of renewal, so it's super exciting for us. So university's definition of an academy is that you come up with programs in research, education, and engagement, but they have to transcend across all 10 colleges. And so that is something that has been a platform for us, is to really engage, break silos, and to really create interdisciplinary environment. So we have very specific programs in research, education, and engagement. One Health is a big umbrella, as you can imagine, that has worked great for us. At the same time, also poses some very interesting challenges in terms of breaking those silos. A key focus for us over the last year or so has been to foster interdisciplinary environment. So Many of these planning events are planned by us, by the core team, by the leadership team, but many of these ideas are really also coming from you. So the reason I'm sharing some of these key highlights is if you have this amazing idea that's stuck inside your head for many years and you have not been able to explore it because you're super busy, don't have the time, give us the opportunity to have a conversation with you. We'd love to talk to you. The only requirement we have from our side is cannot be an N of one. We want to bring in other colleges, other programs to have a discussion with you and to really expand that program. Our affiliate faculty member program has been growing. This is as of a couple of weeks ago. As of this morning, we are on 268 faculty who are affiliates, many from NC State, many from outside. 7 out of the 10 deans at NC State are affiliate members of a program. And I'll welcome you if you have a scan code over there for you to explore. It's a 1-minute Google form that you can easily fill out, but then you get access to a lot of lists of opportunities that we have to offer you. We have done amazing, opportunities to create, more interdisciplinary, seed grants. So this year, our first call, we received around $600,000 in interdisciplinary proposals. We funded around $180,000 for seed grants. We have an open call for faculty travel. And then, of course, we have created multiple working groups. These working groups have been amazing. It gives us an opportunity to learn more about what you do. Just last week, we launched our Global One Health Leaders Seminar Series. We had 4 amazing researchers talking about next-generation molecular diagnostics, which we want to continue to expand and bring more people to the group, at the same time tell people what we are doing at NC State. So these working groups and opportunities led to submission of 4 grants last year with Duke, with UNC, with RTI. The second proposal is with the Duke Medical School, with the UNC Chapel Hill Medical School, a training grant T32. We just got a score for it in our first attempt, so we are super excited about this opportunity. The rest of the proposals are pending. We really are focused on our education program that has been huge for us, and our key focus is to really train future global One Health leaders at NC State. And that extends all the way from undergraduate education to graduate program, to certificate programs, to leadership and entrepreneurship. And the education portfolio is led by Michael Reisky and Kristen Sullivan and Meredith Spence-Beaulieu. So our undergraduate program in Global One Health was already in existence since 2019. As you can see, we get an amazing group of students representing multiple different majors. And again, as we further refresh this program, we are going to launch a nested scholars program in here, maybe around 50 to 20 students that will have tremendous opportunity for experiential learning, maybe within the US, outside the US. Our graduate program, so we get 5 graduate student slots from the provost office. Last year, these are 1-year funding slots. So last year, we received 83 applications. That was the first call we had from 8 colleges. And we funded these amazing 5 students who are Global One Health fellows. This year, we received 63 applications, but we got applications from all 10 colleges at NC State, which I think also exemplifies what really One Health is that big umbrella where people can really find a space for themselves. So we are super excited about it. In addition to that, we have done tiered mentoring program, internships with the museum, worked on certificates, minors, courses. A big tangible outcome for us is to launch a future graduate program in Global One Health. So that's something that we are working with the provost office, with Dr. Helen Chen, with the grad school. Potentially would be a master's program in One Health, a very unique program. We've already been approached by UNC Gillings that they want to think about partnering with us on this particular program. So we are super excited. And then in the engagement space, we are going to be focused on East Africa. We had this big East Africa Symposium in September of last year, and now we are going back to East Africa sometime in June to further, take those partnerships to the next level. We have established a MOU with the Passion Network. This is a network in 5 continents, around 25 different countries, and now we are working on a strategic alignment document with the Passion Network. And then finally, we are working with Colorado State University to launch what is a North America One Health Universities Network. So our hope is that this network will have around 59 to 60 universities in North America engaged in One Health, and so we have already taken a leadership role in there. The first meeting will be in Colorado next year. That meeting will be held at NC State, so that will bring a lot of One Health professionals and folks around the world to NC State. So the final slide is, we are really engaged in global One Health research, education, and engagement model, and to make them seamless and connected to each other, all the way from our 3 research thematic areas that flow into global challenges, into student education, entrepreneurship and innovation, and also, being globally engaged. So big picture, we want to engage with you. We want to learn more about what you're doing, and then what we can do together with you. And all the work is done by this amazing core team that the Academy has. I cannot thank them enough. And then even an amazing leadership group that has representation from every college in that particular group. And many of them are sitting over here, or we're going to hear from them later in the day. Again, thanks, ORI, for the opportunity, and would love to answer any questions. Thanks.
Would anybody like to ask a question? Okay, and so unless anybody wants to ask anything, I would like to get the discussion started because I found these super interesting. And Astrid, do you want to talk a little bit more about how climate change may contribute to toxic blooms and where the blooms end up? Like, where do they go away? Thank you.
That will help. There we go. So yeah, like I said in the introduction, there is some good indication that some of the more toxic strains do better under warmer conditions. There's generally a debate in the harmful algal bloom field whether climate change makes things worse, but there's several regions worldwide where that is actually being documented. So one of the things that makes an additional challenge is each of these different algae have their optimum place, their optimum temperature, where they feel best and where they thrive the most. And with climate change, some of those geographic regions where they do best are also shifting. So you have to also keep an eye on things. For example, the Florida tide has showed up in North Carolina before. It was kind of like in the 80s, where one of the Gulf streams sort of had one of the scale eddies push into the North Carolina estuaries, and that was really bad. We're keeping an eye out on whether that is happening again, but you can imagine as temperature regimes are changing, those algae might go into different places. As far as the fate of these cyanotoxins in freshwater environments goes, there's really very little known. And there's also not a lot of models that have been used, so we're really excited about Scott Belcher's group and Catherine Hoyu trying to tie that to epidemiological liver studies across North Carolina, and then actually looking at some of the combinations of these toxins. So we're studying microcystin is our primary focus, and that's a hepatotoxin, and it has been linked with studies with fatty liver disease and liver toxic effects. And microcystin is not, unfortunately, just one toxin, but it's actually similar to PFAS. It gets complicated. It is dozens of different, actually over 200 different congeners. And what we're trying to do is start with the ones that are most common in the environment and then use those mixtures and look at how they affect liver health. So most studies have been done with fish, zebrafish models, but Scott is going to work with mouse models. So we're hoping to learn more about that.
Yeah, thank you. And so the reason I asked that was because last summer, I happened to be in California when the California sea lions were being affected by these harmful algal blooms, and it was very harmful to the California sea lions. So my next question's for Sid. I can imagine that this interconnection that you were talking about in your last slide is really important when things like this happen. So can you talk a little bit about how that, like a nationwide effort might be able to play into controlling these, like an infectious disease outbreak or something like an algal bloom that's not supposed to be where it is, I guess?
Yes, so infectious disease is huge. So maybe I'll talk a little bit about my own research in the field of antimicrobial resistance. It's a huge global problem right now, and I feel like 2050 is that magical year where everybody starts to bring those estimates. So we have an estimate for, number of deaths caused by antimicrobial resistant pathogens. By the year 2050, it will be 10 million people every year. Cancer will be around 8.2 million people. And you look at the statistics, we are rapidly moving towards that number. The 2019 data published in Lancet in 2022, around 4.2 million deaths were associated globally with AMR pathogens, and 1.4 million were directly attributed to it. And of course, you have to keep in mind the underreporting, reported cases in this field. It's a huge problem, complicated problem, but also one of the best studied One Health model, because you have these pathogens causing massive amounts of deaths in humans. Same thing with the animals, and then there's this big environmental reservoir. So in terms of working together, in the US, we have amazing surveillance programs, but as the pandemic has shown that pathogens can emerge in any part of the world and can be knocking on your doorstep the very next day. So from us, from our perspective, and also from, say, World Health Organization and other areas, really a global effort is needed, and that includes, capacity building efforts, training efforts, because big global problems, don't respect international boundaries. So you can have amazing surveillance programs in the U.S., but they could be, useless the very next day if an emerging strain that's resistant to multiple drugs, enters into your system from some other parts of the world. And so you have these hot spots of emerging strains in different parts of the world. And so a global effort is needed to counteract some of the big problems. And within infectious diseases, AMR is the challenge that is the top 3 concern for most of the world agencies focused on infectious disease control, including CDC, FDA, WHO, and others.
Right. Well, based on that, I mean, COVID wasn't actually on our doorstep. It was kind of knocking down our doors. Do you have any hopeful, optimistic views that you want to share for the coming years?
Yeah, AMR is mostly doom and gloom type, right? No, I think at the global level, I'm really encouraged to see some of the steps, especially taken in One Health. So, the quadripartite One Health agreement signed by WHO, FAO, World Animal Organization, and UN Environmental Program that has 6 different action tracks, very focused on One Health. And most of the action tracks are very big picture, but action track number 5 is focused on antimicrobial resistance just because it's a big concern. But if you look at that agreement, it's very focused on working together. It's almost a theory of change model. And the concerns are the same, but the approach is more One Health in nature, right? And so that really, makes me feel more, much better, state as compared to others. Because for these big global programs, you really have to bring in people from different skill sets. Yeah. One person with one skill set cannot solve it. So seeing these big efforts being made at the global level is really gives me a lot of enthusiasm that we're going to be working on it in a much more concerted effort.
Right. And speaking of working together, my next question's for Astrid. So I think, a lot of people may understand that some of the chemicals maybe that I talked about have effects on liver disease or metabolic disease. And so now you and some of your new collaborators are going to be actually looking at the effects of microcystins on liver, how it's contributing. Because I think we all understand now that we're not all exposed to one chemical at a time. It's a mixture, right? Mm-hmm. But do you want to talk a little bit more about how, the new things that have really come up, that the new things that you think you can tackle with the OSHA and Human Health Center?
Sure. So just as a disclaimer, we just got funded, but
optimistically.
Yeah, so some of the things that are big question marks is actually what the environmental relevant concentrations are, what the spatiotemporal patterns are. If you look for cyanotoxins in the environment, you will find them. This is not unique. It's never been considered a problem in North Carolina, so you've probably heard about the Toledo water crisis, the Great Lakes, Lake Okeechobee and whatnot. In 2019, we found environmental concentrations that were 3 orders of magnitude higher than the Toledo crisis that shut down the water treatment plants. So I think what the center really allows you is the connection between getting relevant concentrations in the environment, not only there, but also in oysters and blue crab, that can inform the biomedical studies, the trials with your mouse models, and then that feeds into the models that our modeling group is looking at, and you can get these connections with climate change. So it's not just that we're producing brand new data, we're also allowing those to be incorporated in looking at the health implications, and then into models that can connect with communities that have those questions. They have their peers right into the children or into those environments, and when it's green, they wonder if their kids can jump in, right? So these are really relevant questions. Just as a disclaimer, 2019 was really bad. Our lab is studying all sorts of, like, you name it, blue catfish, white perch. I don't know how many fisher people are in here. We do find it, but typically only in viscera, gut, and liver, and not necessarily making it into the fillet. So keep on preparing those things the proper way. But what we're seeing is that every now and then it gets worse. And what we want to do is we want to tell people, okay, stay away from this animal or that consumption type for that type of amount. So not everything is bad. There is, like, going back to positive things, but you can put information that is targeted to the situation and to the water uses, and then warn people off for a little while, but then come back and use those resources. We don't want to, I'm not an oyster fan, but I'm a fan of any other type of seafood, so I'm still eating it. Especially if I have this kind of information. So I think this is what the center can help do, and that's really exciting.
Yeah. All right, well, with that, we are out of time, and I want to thank you so much, and thank you all for your attention.
Thank you very much to our panelists and Sue for moderating. We really appreciate you bringing this very relevant topic to our group today. It is everyone's favorite time today. It's lunch. So we do have a catered lunch buffet. It's been set up towards your right and my left of the room. Note there are 2 sides to the table, so please form 2 lines, utilize both sides. And then, so we're gonna ask you to grab your plates and then try to return to your seats by 12:20. We do have a special presentation today over lunch. So while you eat, our special presentation for the audience will be brought to you by members of our proposal development team unit and our foundations team regarding the research landscape and funding opportunities in the area of 21st century threats to health. So we'll go ahead and form 2 lines over here for the lunch buffet and get that started, and then come back to your seats when you've had a chance to do a bio break, and we'll get started with the special presentation.
Hey, everybody, I know you're eating. I'm going to take a minute just to interrupt you. I get the privilege of doing the easy job of saying a couple thank yous that I'd like to say. First, I want to thank everybody for being here today and all the speakers. We've had some excellent presentations so far today. So I first want to thank you for taking the time out of your days. I know this is a big ask to take out of your calendar and spend the time here today. So that's my first thank you to everybody here. The second one, while we have this nice crowd and I've got your mouths full of food, I want to make sure we take the time to thank our committee, our planning committee. As you know, symposiums like this take a lot of time to plan, and we've already have RLA members that are incredibly busy. As you know, the Research Leadership Academy is made up of faculty members that are incredibly busy conducting research and mentoring, and so I'd like to acknowledge the committee members. We have Heike Sederoff, who's our chair, as you know. We have Jacob Jones, who's sitting over here, Sylvia Nassar, who I don't think is able to be here today, and Aaron Sills, who I think will be joining us this afternoon. So I want to acknowledge those members who helped plan. And then I, of course, have to also acknowledge the ORI Planning Committee with Laura Kroger, who we all know, keeps us all moving and keeps us looking professional, and Sophia, who's at the back, one of our newer members, who I know you'll all get to know much better. So I first wanted to make sure I said that. And then we have a special thank you. So finish chewing, Heather. Okay. So one last one that's not quite as happy a thank you, but I'll try to stay happy up here. Many of you know that this is Heather's last University Research Symposium, and that she'll be leaving NC State here soon, and that she's been the chair of the Research Leadership Academy. And she's going to be leaving us to pursue the next stage of her career as the Director of Translational Toxicology at the National Institute of Environmental Health Services. I almost said sciences. It is sciences. You wrote services. Not my fault. Sorry, that's okay. I want to acknowledge Heather's contribution here to NC State and the Research Leadership Academy and the research community. She received the Outstanding Research Award and was inducted into the Research Leadership Academy in 2019 and became the Academy's chair-elect during the 22-23 academic year. And last year, she was the planning chair for our URS, right? So she knows a little bit about what it takes to plan this. And that symposium was focused on the topic of putting data sciences into practice, emerging roles for AI in research. And so hopefully some of you attended that one. Heather, we are really sad to see you go, but you've been incredible as a leader here, not only at NC State, obviously in your College of Sciences as well, and in the research community. And we wish you well in your next adventure. But we have a couple of things we just want to show you some appreciation. If you'll come up here, we'd really appreciate it. Yes, we really appreciate your leadership and time. It takes a lot of additional effort to serve the research community and serve as co-chair and chair, and so we really do want to thank you for your time.
Thank you so much. The best thing about NC State is its people, and the people in this room are some of the most talented, amazing, generous, innovative people that I've met, and I look forward in my new role to just strengthening relationships between NIH and NC State, because go Pack.
Yeah, congratulations. All right, thank you, everyone.
We also, as I mentioned earlier, have a special presentation while you're dining. And at this time, I'd like to ask our next presenters to come up to the stage. So today we will have Michelle Frick, our Senior Proposal Developer in our Proposal Development Unit, and Samara Hanna, the Executive Director for Corporate and Foundation Relations. They are going to give us a little bit of 21st century threats to health funding and collaboration opportunities. So ask you all to come up.
situated here. All right, so today we wanted to talk a little bit about and present some relevant funding and collaboration opportunities kind of in this vein that we've been talking about, 21st century threats to health. And my colleague Molly Puente was supposed to be with me today, but she's not feeling well, so I'm going to take the reins for the proposal development unit, but then I'm going to turn things over to Samara to talk a little bit about corporate and foundation relations and how we work together and how you can kind of utilize both of our services. So that being said, I'll say a little bit about the proposal development unit if you don't know who we are. We provide assistance to faculty across the university on usually multidisciplinary proposals that are over $1 million. You can request our services on our website, and there's a bunch of other things on our website too. You can get access to our newsletter and recordings of all our past webinars and workshops. Some funding agency guidelines. We have pages about NSF and USDA and DoD, et cetera, and then some other links to grant writing resources at NC State. So before I get into some of the relevant funding opportunities we pulled to talk to you about today, you have to kind of think about them in perspective of the challenges that you face when you're going after federal funding, especially in this area. And so a few things we thought about were navigating the slow pace of government funding with the need to be nimble, understanding mission boundaries, so where one agency stops and the next picks up and what's most relevant for the type of work that you want to do, and then scaling your research to meet the size of the threat, but also the size of the funding available, right? And so with all of these couple of challenges in mind, we wanted to present a handful of relevant funding opportunities by some of the agencies that most typically fund this sort of work. So first I'll start with the National Science Foundation, NSF. The first one up here, and I guess I should stop and say, all of these, we've put the deadlines, the upcoming current deadlines for these opportunities. But I think everything I'm going to present to you today is reoccurring. So while you may not be able to go after a cyber physical systems proposal right now by May, it will come back up and around. So all of these are kind of not just one-offs. But the first proposal opportunity, the cyber physical systems or CPS proposals, these are great for teams interested in exploring engineered systems with computational and physical components. Proposals need to develop core research to engineer these complex CPS systems and close the loop, so to speak. This program is looking for cross-cutting fundamental scientific and engineering principles, and it's great for programs for teams interested in domains like aeronautics, agriculture, manufacturing and transportation, energy, and health. The next one, building synthetic microbial communities. This proposal opportunity is great for teams interested in the convergence of biology and engineering, and in addressing fundamental questions in biological sciences and pressing societal goals such as mitigating the effects of climate change, biodegradation of forever chemicals, sustainable crop production, and in correcting imbalances in microbial populations. I guess I should also say, as you all were talking this morning, it was exciting to see that a lot of the things that we chose to present to you today were things that a lot of the teams have been talking about. So they seem really relevant to the work that you all are doing. The next one, smart health and biomedical research. This proposal opportunity would be great for interdisciplinary teams interested in advancing fundamental science and engineering by addressing pressing questions in the biomedical and public health communities. Projects should develop high-risk, high-reward advances in computer and information science, engineering, mathematics, statistics, behavioral, and/or cognitive research. The next one, organismal response to climate change, will be great for teams interested in use-inspired applications of foundational research and in convergence research to develop integrative cross-disciplinary approaches. Specific areas of emphasis include integrating physiology and genomics into the next generation of species distribution models, understanding the mechanistic basis of plastic responses to climate change, functional genomics of organismal response to climate change, and improving our ability to predict the limits of biological and global resistance as organisms face changing and novel climate conditions. The next plant genome research proposal opportunity is great for teams interested in interdisciplinary research on a genome-wide scale to provide new insights into plant processes that have the potential to advance food and national security and contribute to the bioeconomy. This program has a broad scope, and supports studies of plants across the kingdom. And they have an emphasis on the scale and depth of the question that's being addressed and the creativity of the proposed approach. And then the last NSF one we wanted to just mention is the rapid mechanism for proposals. So this falls into the other type of proposals category at NSF, and it's used to address a severe urgency in the ability of or access to data, facilities, or specialized equipment, including quick response research on natural or anthropogenic events and similar unanticipated occurrences. And so because the focus on urgency and quick response research, this would be highly appropriate for when you're thinking about 21st centuries to health. The next organization is USDA, which I work a lot with. A lot of the proposals I work on are in the Plant Science Initiative. So the first example here, Nutrition Incentive Program, funds projects aiming to increase the purchase of fruits and vegetables among SNAP participants by providing incentives at the point of purchase. So this is great for teams interested in bringing together stakeholders from various parts of the food and healthcare systems. And there's a huge emphasis of this proposal opportunity with evaluation. So you have to have a strong evaluation component for this particular opportunity. The next one, the Genome to Phenome Initiative, is best for interdisciplinary research teams, which you probably noticed I've said interdisciplinary almost every time. With an interest in data or data infrastructure, the program focuses on collaborative science engagement to develop a community of researchers across both crops and animals that will lay the foundation for expanding knowledge concerning genomes and phenomes of importance to U.S. agriculture. Next, the SAS opportunity, which recently came out for this year. SAS proposals are complex. They are great for large interdisciplinary teams interested in transdisciplinary science through close collaborations with both researchers and stakeholders. Projects have to focus on systems approaches that significantly improve the supply of climate-smart, sustainable, safe, healthy, and accessible food and other ag products. Successful projects are well integrated, so they have to have research components, education components, and extension components to be successful. And they require a meaningful collaboration with the stakeholder community. And then the last USDA proposal we wanted to mention is the FAST, or the Foundational and Applied Science Program, which supports grants in priority areas ranging from plant health production and products to animal health production and products, food safety, nutrition, bioenergy, and agriculture, economics, and rural communities. This RFP is unique to the other ones on this list because there are a number of program areas within this one RFP. Each program area has specific project goals, so it can serve a wide variety of project teams. There are foundational and applied projects in this RFP, as well as different project types. So some are research only, some are education only, some are extension, and then some are integrated. And so we thought this would be a good opportunity to give you a little plug for our next PDU at Lunch, which is going to be on April 10th. And myself and a colleague in the Plant Science Initiative are going to talk about best practices for including extension services in your research proposal. And so this is a great opportunity both for USDA proposals, which you probably think about extension the most in the ag space, but it can also help impact your NSF proposals and their broader impacts because there are extension agents in every county in North Carolina. So you can reach any group of community stakeholders. It doesn't just have to be farmers or producers. You can really get involved with the community in whichever area, whichever part of the state that you're wanting to connect with. So we're going to talk a little bit about that on April 10th. You can scan this QR code to register. You can go on our website, and there's a link there as well. Wanted to talk just briefly about USAID because they're a very interesting and very different agency than a lot of the other ones you're probably used to working with if you don't know a lot about USAID. But USAID funds proposals for international development and humanitarian efforts to promote global health, support global stability, provide humanitarian assistance, and catalyze innovation and partnership. USAID has a number of focus areas, including some that are directly related to what we're talking about today, such as agriculture and food security, environment, energy and infrastructure, global health, and water and sanitation, among many others. They do take unsolicited proposals, so we wanted to mention those, because if there is not a relevant opportunity with USAID that kind of seems to fit your need, they will take unsolicited. You have to have 3 pieces of criteria, though, to get through it, and they do only fund a small number of these, so just to make that clear. But they will accept unsolicited proposals that clearly demonstrate a unique, innovative, or proprietary program, that show USAID support would be worthwhile, and that the proposed work could benefit USAID's research and development, and that fit within an existing USAID objective. So you can go online on their website on USAID and find all the information about how to submit an unsolicited proposal, but it involves submitting a concept paper to kind of see the fit and make sure that it is suitable for what their program is, and then you can get to the next stage of submitting a full proposal. But the majority of USAID proposals are solicited, so kind of the list of the rest of these. Many are country-specific, like these here, and they are all over the globe. So just because, South Africa, Vietnam, the Philippines, these are the ones that are listed here, doesn't mean these are the only countries that they're in. USAID proposals are great for teams that want to do international work, since a lot of the other federal agencies are so strict about working with international partners. And then next, of course, we have to talk about NIH if we're talking about 21st century threats to health. So the first one listed here, this notice of special interest for stimulating research to understanding and address hunger, food, and nutrition insecurity. This would be great for teams interested in looking at food and nutrition insecurity and how that affects a variety of health outcomes. This program is interested in development and testing of interventions, innovative programs, and natural experiments to address food insecurity and neighborhood-level access to healthy and affordable foods. The next one, risk and protective factors of family health and family-level interventions. This program recognizes that research on patient populations has centered on the health of the individual or the patient level rather than, while ignoring the health and well-being of other family members or on the family health unit. So this opportunity seeks multidisciplinary research on minority health and health disparities and interventions at the interpersonal level, especially within and at the family level. The next one, Exploratory Developmental Research Grant Program. This is out of the National Institute on Minority Health and Health Disparities, and they're looking for new exploratory or developmental research projects that have the potential to break new ground in the fields of minority health and health disparities, or extend previous discoveries toward new directions or applications that can directly contribute to improving minority health or reducing health disparities. And then the final NIH one, this time-sensitive research opportunities RFP, it reminds me of NSF's RAPID a little bit in that it's quick and urgent. So this is for environmental health science teams who want to respond to an immediate event. And this opportunity supports research in environmental health science in which an event has or will imminently occur that provides a limited window of opportunity to collect samples and data to support the assessment of exposures and human health impact. The distinguishing feature of this opportunity is that the turnaround is substantially shorter than the typical NIH grant process, so it should be clear that there's a limited opportunity to address this unique and important research, and investigators have to describe their capacity to begin a project right upon award. And then finally, before I turn it over, we would be remiss if we didn't mention some of the other federal agencies that are out there. I think, you think about NIH, you think about NSF, you think about USDA potentially, but there are other agencies that will fund this type of research. So we just listed a few here, the U.S. Department of Homeland Security, the Health Resources and Service Administration, and then the newly formed ARPA-H are all really great places to look for if you're looking for research opportunities in this landscape.
Thanks, Michelle, so much. I'm Samara Hanna. I'm Executive Director for Corporate and Foundation Relations. And my presentation is going to be a little bit different. I really want to make sure that I give everyone an opportunity to really understand what corporate and foundation relations is here at NC State and how our program is similar and aligns with PDU, but also how we're a little bit different. So first, we are located centrally. We're part of university development. And we support all the units across the university. So we don't report to a dean. We don't report to any of the individual units. So one of the things that makes us different is that we can work across the entire university, or I guess it makes us the same as PDU, but it makes us different than our colleagues in the units. So every unit has a chief development officer, a chief fundraiser. We work collaboratively with them, but we try to manage those relationships that can cut across the university. We look for proactive strategies that help us to focus on the priorities of NC State, but we also work responsibly and we acknowledge the RFPs and we try to find ways to secure those funds on behalf of the university while also looking more proactively. So sort of an example of that is we're working with some colleagues right now who are anticipating a federal grant that we'll hear about probably in the next 3 months. That federal grant does not require any matching opportunities, but if there is a match, there's more money that comes for that. So we're starting to look right now to line up those matching dollars to align with that federal funding, should we get it, to then secure additional monies on top of that. So that's one way that my office can certainly be helpful and help align with the needs and priorities across the university. And then we do look for the larger opportunities. So a lot of folks need help at all different levels. Because we're such a small office, we do have to look at the largest opportunities possible. It doesn't mean that we're not available to help people where they need help, but we do have to prioritize our resources and look for those relationships. I call them and relationships. So Michelle and I talk all the time. If she's working with a foundation and I'm working with a foundation, we definitely talk about what can we do to expand that relationship and make it bigger within the boundaries of NC State. Did that advance up there? Yeah. Okay, good. So we work with our top corporations, corporate foundations, private foundations, and we build strategic relationships that help to enable that interactive relationship between the university and the organization that we work with. We serve as a connector to facilitate mutually beneficial relationships that are not only, as I said, responsive, but also proactive. So much like the gift matching opportunity that we're looking for, we're outreaching to these organizations and trying to align that funding right now. And while my shop's priority is philanthropic support for the university, we're not exclusive to that. And that's where relationships with programs like PDU come in, because as we find those places that are not purely philanthropic, we do reach out to our partners on campus and engage them in the conversation as well. I want to talk a little bit about some giving trends that we're seeing right now within the landscape of the United States. So last year, corporate giving was up. There was about $30 billion that was given across through all companies. And the priorities, of course, sustainability, disaster relief, crisis relief, I think we all are starting to see that trend continue. There's a bigger focus and a growing focus in sort of giving back to local communities where companies have a presence. They're also listening more to what their employees' interests are. So that's where our alumni are really important to us. We've got to nurture those relationships every single day. And then companies are looking for developing partnerships. They don't necessarily want to just give funding to an organization. They're looking to establish a partnership. So as we have those conversations with these organizations, they're looking to make sure that, if we're asking them for $1 million or a couple $1 million, what is that partnership going to look like? And there's just got to be a lot of conversation within that. As we look into this year, what we're starting to see in trends, obviously it's an election year, so that's going to have some impact. The economy has been a huge driving factor for where companies are starting to give their money, and a lot of foundations as well. Most foundations have endowments, and the way their endowments work, they prioritize not only their giving, but also sort of the more basic needs in times of crisis tend to get funded over things that we would generally look for here at the university. So we see some shifting in funding within the university. More things like PAC Essentials are getting more attention as opposed to traditional things like scholarships and whatnot. Companies are monitoring the role of artificial intelligence and we don't know what the full impact is right now on philanthropy, but that is definitely having an impact on philanthropy. And then, of course, back to that employee interest piece, generational preferences. So as companies are talking to their employees, what do those employees want to see the company prioritizing as far as their corporate responsibility goes, and how do those funds get distributed? And I am going to give a couple of examples. I'm not going to drill down quite as deeply as Michelle did, but just to give you a few examples of how our office works. And Keck is a great example because we do this directly in collaboration with the Proposal Development Unit. But this is a really wonderful example of how our offices work together. So the Keck Foundation requires that someone from my office or similar office be their point person. They don't want to talk to a researcher. They don't want to talk to a PI. They actually say that it has to be someone who comes from the giving office, and it's typically a corporate foundation relations officer who works with them specifically. But there is an open RFP right now for Keck, and if you all are interested, I would encourage you to either check out the Keck website or check out the PDU website because it is on there, and we do facilitate our application through the PDU office. Burroughs Welcome Fund, that's another one. I think most people in this room have probably tried to get funding, have secured funding, or are in conversations with me about getting funding. But Burroughs Welcome Fund is a great conversation to have because there are so many opportunities with them. And it's a really good example of how we can have multiple conversations going with a major funder at any given time. And to have someone like me as a point person really not only facilitates the conversation for us, but it facilitates the conversation for them so that we're not sending them mixed messages or bombarding them or overwhelming them, I guess, with conflicting funding requests or just, sort of too much coming at them at one time with putting ourselves in a competing situation with them. And then the last example I want to share is the Z. Smith Reynolds Foundation. So this is another one that really has strict guidelines, and we've got to be very deliberate about how we apply within the boundaries of what it is that they do. But they really appreciate the opportunity to work with NC State. They have a fairly new executive director. I think she's been there for about a year now, and she's super excited about working with us and continuing to support us. But these are just 3 examples of how having those relationships with a point person within a central unit allows us to engage these relationships across the entire university and keep positioning us as the leadership in these organizations continue to change. It keeps us at the forefront of their funding conscience stream of mind as well. And then my last slide is just a couple of examples. I talked earlier about how we have to prioritize our resources and really think about some of the larger opportunities out there. This is not an exhaustive list by any means. We work with about 100 organizations within my department. But I just wanted to give you some examples so that you had a really good sense of how it is that we work. And you'll notice a lot of these also align with the lists that you see on the PDU website, also on the lists and the conversations you have with folks in many, many other programs. But what my office tries to do is take a look at what I call our top 100. That's not a perfect number. But we definitely want to look at those top 100, find out what they're doing across NC State, and how do we take those relationships and really nurture them in a way that makes sure we are maximizing the opportunity. And not necessarily to move them. If they're currently funding the College of Engineering, we don't want to move that, diminish it in any way. We want to add to that. Perhaps there's an opportunity to get them engaged with someone in the Poole College of Management or in CALS or somewhere else. We just need to be able to cross those conversations throughout the university, and that's where a central conversation between my office and colleagues in PDU is really important. And that is the end of the Corporate and Foundation Relations 101 conversation. I will just finally say that when we grow up, because we are a small and a much younger part of the NC State fabric here, when we grow up, I want to be just like PDU. Thank you.
Questions, comments, concerns?
No concerns is good.
I know.
All right, thank you.
Thank you, Michelle and Samara. That was great information for this group to know. We are going to give you 15 more minutes, so we're not going to start our next section until about 1:15. So take this opportunity to get another plate of food, maybe connect with somebody in the room and take a bio break and refill your drinks. We will be back at 1:15 with our next set of highlighted speakers for today.
Okay, hello everybody. I hope you enjoyed the lunch and the PDU presentations. And I hope you had a little coffee or tea, something to keep you awake, because we have two really interesting new highlighted speakers in this session. And we focused this session a little bit more on social science and risk assessment research, so not at the hardcore natural science research, but really going outside. And we have two really amazing speakers. First one is Dr. Kara Grieger. She is an associate professor in applied ecology and an expert in environmental health risk assessment, something we need. She also is a Goodnight Early Career Innovator awardee, and a member of the STEP Center. You remember the first presentation this morning from Jacob Jones, who is your boss there. So Kara's work, as I said, focuses on the best practices for risk assessment, risk management, and stakeholder engagement. So she's the one talking to everybody, related to governing and emerging risks of new technologies.
Thank you so much for having me here. I assume I just click here, and maybe this one. There we go. All right. I think one more. All right, there we go. Good afternoon, everyone. I'm really excited to be here today with all of you to share some information on interdisciplinary approaches and responsible innovation for developing real-world solutions. All right, so as background, and I don't think I have to spend a lot of time talking about this, it's becoming increasingly clear that interdisciplinary solutions are urgently needed to ensure resilient and sustainable environments, and this includes the development of safe and secure food and ag systems. So this includes, for example, developing new technologies or new and better crop varieties to ensuring adequate food safety systems are in place, as well as fostering equitable food distribution and access. And often is a critical part of these solutions to developing more sustainable futures is the key role of new technologies in innovation. In the case of developing more sustainable food and ag systems, the field of nanotechnology and genetic engineering may hold great promise. For example, new nanoscale fertilizers and pesticides may serve as more efficient mechanisms to deliver agrochemicals with potentially less runoff compared to conventional approaches. And new and novel applications of genetic engineering in both plants and animals may offer products that increase animal welfare, greater productivity, better tasting and more nutritious foods, and reduce food loss in some cases. But at the same time, we also know that new technologies and innovations can present new or unknown risks in of themselves. Actually, because new technologies are inherently new and novel, it takes time to collect information about their potential risks and impacts, and even more time to then make decisions regarding those risks and impacts. And so this lag time between technology innovation, understanding its risk and impacts, and then being able to make decisions is called the pacing problem, and we've been dealing with this for decades, actually. So in the near term, we're often operating in this space of very deep and varied uncertainties. Actually, right now, we have a lot of well-known examples of what I'm talking about here with the pacing problem. So some really, bad and clear examples are things like asbestos, once heralded as a magic mineral, DDT, first generation of genetically modified organisms, in particular their use in crops. More recently, we all experienced some of the societal concerns regarding new COVID vaccines, emerging health and environmental risks of PFAS contamination, which we heard about earlier from the CHHE director, and societal and ethical concerns regarding the growing use of artificial intelligence. So all of these examples that I'm trying to show you illustrate how new technologies and innovation can indeed be very promising, but they can also present new and unknown risks and risks not just to human health, but also to the environment as well as society. So there's many different types of risks that we need to consider. Okay, so as a way to develop more sustainable food and ag systems, and particularly those that rely on new and novel technologies, we need to better incorporate interdisciplinary approaches right in the beginning of research and innovation cycles. So we need approaches that will tap into diverse disciplines and perspectives, drawing from a large number of fields like social sciences, natural sciences, engineering, policy, and beyond, while also ensuring diversity, equity, inclusion, and justice. This is also important to do so because many of our complex environmental and societal problems, like phosphorus, like chemical contamination, like the growing concerns over artificial intelligence, are wicked challenges where there may be difficulties in even defining the problem. There's no single bullet solution. They can have open-ended timeframes. They may be new and novel in themselves, and there can be competing values or objectives between stakeholders. So, if we can go. For these reasons, my lab here at NC State, called the Interdisciplinary Risk Sciences Group, is currently working on a number of externally funded grants where we're trying to understand some of the social and societal implications that explore different dimensions of human health, environmental, and socio-ethical considerations. We tried to also evaluate benefits and risks of novel agri-food products that rely on new technologies. We also try to understand stakeholder perceptions and needs regarding these technologies, and overall try to support concepts of responsible innovation. So this is what we're doing in our lab. One of the case studies I wanted to pull out from our giant toolbox, I would say, that kind of describes some of the work that we're doing involves the use of nanotechnology in food and agriculture. So as background, nanotechnology actually refers to the field of science and engineering where we can manipulate, control, and engineer materials really at the nanoscale. So this is around approximately 1 to 100 nanometers. So at this very small atomic scale, materials can actually have very different physical and chemical properties. Jacob Jones can probably tell you more about those properties as a material scientist. So scientists and engineers have been really interested for the past couple decades on using different nanoscale materials in various applications, including food and ag systems. So starting around the mid-2000s, the field of nanotechnology was launched and incorporated in with the strategic launch of the field of nanotechnology. Incorporated deep into this was a core principle of its responsible development right from the beginning. And why responsible development was integrated with nanotechnology strategic plans was because of that public backlash that we experienced with the first generation of GMOs. So nanotechnology researchers and scientists, they were like, wow, I don't want to go through that. You see what just happened? I don't want to go through that with nano. So they particularly put the field of responsible development in its strategic plan from the beginning. I was actually trained as an environmental engineer. And so I started launching into understanding understanding the different environmental risks of nanotechnology and engineered nanomaterials, looking at different applications of how we use nano in the environment. But after about 8 years of conducting those types of research, I began exploring potential risks and aspects of responsible development of nanotechnology in food and agriculture. So starting around 2016, colleagues and I started to investigate the use of different nanomaterials in food and how they have different fate and transformation in our bodies, as well as looking at some of the evolving literature on potential impacts. We also started to investigate some of the analytical challenges to even being able to detect, characterize, and quantify the use of different nanomaterials in food, especially because in Europe, European regulators decided that it was time to label foods that contain engineered nanomaterials on them. So today, if you go to Europe and you buy a pack of gum, you will see titanium dioxide, parentheses, nano, because of a mandated food labeling law in Europe. So here, I started, after understanding more of these technical aspects, I began began being interested in some more of the societal and ethical dimensions of the use of nanomaterials in food and agriculture products. So through funding provided by the USDA, colleagues and I wanted to understand everybody wants to do responsible development, or it's at least in our strategic plans, but how has this been done for nano in food and ag? If researchers are wanting to do this, are they running into any barriers? And if so, what are those barriers? And overall, how can we foster a better responsible innovation of nanotechnology, particularly in food and ag, as we go forward? So obviously, I don't have time to share the whole plethora of research findings from this work, but I will share just a few insights here. So we conducted a number of interviews with researchers in the US that were developing nanotechnology in food and agriculture products. And we were understanding how they viewed responsible innovation, what they were doing in their own labs to responsibly innovate. And along this way, we found that there were a number of barriers that they were running into. The first barrier that they were running into to responsibly innovate was the lack of data. So they didn't have enough information or data about the impact of their product on health, the environment, society. Was it going to be accepted? Again, back to that pacing problem. They also talked about the lack of product oversight or regulation. They were either missing or unclear regulations for them to follow in some cases. They also talked about the need to still be able to market and sell their product at the end of the day. Yes, they were interested in responsibly developing their product, but they also needed to sell it and make sure that they could make a profit at the end of the day. They talked about the need for increased collaborations. In many cases, they need to collaborate with others to responsibly innovate. And finally, they talked about the need for having a more adequate training and workforce. They needed their new postdocs and scientists to be more comprehensively trained so they could responsibly innovate. So the story is we want to do responsible innovation of nano and food and ag, but we're running into some of these challenges. Next, in part of the study, we engaged different expert stakeholders using an online platform. So this is during COVID. We wanted to understand what stakeholders perceived about nanotechnology and food and ag products and what they thought researchers and innovators could do to better responsibly innovate. So among other activities that we conducted, participants reviewed 5 different case studies of using nanotechnology in food and ag, including nano-sized emulsions that would coat fresh fruit to prevent browning, nano-encased vaccines for hens to protect against salmonella, dietary supplements of copper for human consumption, nano-zinc oxide to prevent citrus greening, and nano-titanium dioxide in infant formula, so it's more white. So some of these were for nano in food directly that you consume, some were for nano in ag. What we found when we asked these stakeholders, please review the case studies, tell us how responsibly innovative that they were. We found that the higher bars were really those with nanotechnology in agriculture. So overall, we were finding that stakeholders considered nano in agriculture products to be more responsibly innovated than those directly in food, where the most responsibly innovated case study was that nano zinc oxide to prevent citrus greening, and the least responsibly innovated case study was nano titanium dioxide in infant formula. And then we asked, well, why did you rank them according to these degrees of responsible innovation? And the main reasons, there were several reasons, but I'm going to show you the top 3. The main reasons were due to issues of safety. They were concerned about some of the safety implications of these nanomaterials in these products. And secondly, and I think this is very important to highlight, they talked about whether the product addressed a societal problem or need. Many of the stakeholders talked about that they didn't want just another nanotechnology product for the sake of it, or the company could, sell something new. They really wanted products that would respond to a need that we have in society. And then third, they talked about this risk-benefit comparison. Maybe you're willing to take a risk if the benefit is high, but if the risk is high and the benefit is low, then that doesn't sound, very responsibly innovated. So this makes a lot of sense. Then when we asked these stakeholders, okay, do you have recommendations for people developing these materials? So nano researchers and innovators, what should they be doing to more responsibly innovate in your view? And the top responses were very similarly, that more studies on environmental health and safety should be conducted, that They wanted those researchers to make sure their product addresses a societal need. They wanted more engagement with stakeholders and the public. They wanted increased transparency. So you see, safety, science studies is the top, but other than that, there's all socially relevant items that could be done by researchers and innovators to more responsibly innovate. And this has relevancy not just for nano, but for other emerging technologies in food and ag as well. Okay, so we took these findings and we wrapped them up in some best practices. And I'll go through these relatively quickly. Our best practices are that if you are interested in developing new technologies in food and ag, what could you do to more responsibly innovate? Well, you could try to embed mechanisms of responsible innovation in your lab that go beyond safety, that go more towards stakeholder engagement, transparency. We can train our new generation of students and postdocs to do some of this work, again, that goes beyond just the safety, focuses on transparency, trust issues. We don't need to apply the same degree of responsible innovation to all new products. You can have a tiered approach depending on the case, and we need more systems in place that would monitor whether our responsible innovation practices are working. So I'm gonna close up in just a couple of slides, but so based on these findings, we were actually able to secure another set of funding through USDA that focuses on nano as well as genetic engineering in food and ag, and that's the current work that we're working on now, is to try to understand the societal implications of these sets of technologies in food and ag, looking at stakeholder perceptions and needs. I'm gonna skip some over these. But just to say and wrap up that I hope today I've shared with you how interdisciplinary solutions are needed to ensure resilient and sustainable environments, that again, new technologies can hold great promise, but they can also present some new or unforeseen risks. And so to better reduce risks, align with societal values, needs, and expectations, there's an opportunity here to support practices of responsible innovation, not only to conduct safety studies, but also to do things like engage stakeholders in technology design and innovation right from the beginning, understand their needs, understand their concerns, and innovate around that. And finally, I tried to provide an example with nano in food and ag, and we have some more work with genetic engineering as well. So thank you so much for your patience, and we'll have to answer any questions.
In considering the overall spectrum of risk assessment and effects in our society, what is the military doing? And what do we know whether they're a significant factor in our view of risk assessment?
If I understood you correctly, you said, given basically what is the military doing? And does that factor into our risk assessment processes? I'm not sure I can speak to that directly. I do have colleagues in the DoD that are actually looking at risk and making risk-based decisions. But I think there are many different agencies in, the military or DoD. But I can't speak to the specifics of that.
Congratulations on your new project.
Thank you.
You're welcome. So how do you think about the precautionary principle with your work?
Thank you for asking that question. So I spent about a decade in Denmark, and when I was studying and doing my postdoc there on risks and emerging technologies, it was within a European system where the precautionary principle was embedded. So in the case of uncertainty, then the idea is you side on, you err on the side of precaution. We don't necessarily have that same system here in the United States. So I will say, we have what is called a more science-based system, but the problem with that is that lag time that I referred to earlier, that it can often take decades, literally decades, before we have enough evidence to make a regulatory decision. We're dealing with that right now with PFAS. We've known about PFAS for a long time, and just recently, just last year, I think it was last year, just recently, the EPA has proposed some new MCLs for PFAS in public drinking water. So we are dealing with this lag time. There's a lot of discussion around that. There's value-based decisions around that. That's not our current approach that we're using here.
Thank you, Cara. That was excellent. Our next speaker in this highlighted series is Dr. Andrew Beiner. Andy is an associate professor in the Department of Communication and is the director of the graduate programs in CHAS. His research is on controversial science topics and how to including how information flows through different science channels, transmitted through channels, and what impact that communication has on risk perception and public understanding of science. So really important for many of us. His topic today is knowing, feeling, and believing, the role of environmental health literacy to overcome health disparities. Sorry. Thank you, Andy.
Thank you, Heike, for the introduction. Thank you again for the invitation to come present. Good afternoon, everybody. No, it's good. I'm pleased to be here as, I believe, one of the only representatives from the College of Humanities and Social Sciences, so shout out to the Humanities and Social Sciences. As Heike mentioned, I'm an associate professor in communication. I'm also the director or the minor coordinator for our undergraduate minor in science communication. So some of you may not be aware of that. If you have talented undergraduate students that you work with, I'd highly recommend that you direct them towards me in case they're interested in minoring in science communication. It's been a very successful program since about 2020. Today I'm going to talk to you about communication and environmental health. I'm going to share with you some results from a particular article that we published a couple years ago. But first I wanted to let you know a little bit more about me. I am the co-director of the Community Engagement Core at the Center for Human Health and the Environment since 2018. You heard earlier from our new director, Sue Fenton, on that center and all the important work we're doing. So I'm the co-director along with my colleague, Katie May. I have an interest in environmental health that goes back to my time in the Peace Corps when I worked in Central Africa in Cameroon. That's where I really got interested in sort of how people absorb information about risks they face in their lives and what they do about it. And so that led to my research on how people use mass communication in particular, how they make sense of risk in their lives, and how science plays a role in that process. So for this study, we are focusing on environmental health literacy. So why would we do that? Environmental health literacy basically gives us an opportunity, it's a concept that gives us an opportunity to understand and address community health threats. And so there's a lot of research that sort of focuses on environmental health literacy with a priority on local context and local knowledge, right? So going into communities and sort of helping them get resources, information, financial resources that they need to combat environmental health risks. And there's also research that measures this concept through standard survey questions, but sort of leaving out that local context and local knowledge. And so with the study I'm talking to you about today, we wanted to sort of combine those disparate approaches, right? We wanted an approach that acknowledges the importance of local context and knowledge. And so we're focusing on North Carolina as sort of a test case, but that it also may be generalized to larger populations. So trying to sort of thread that needle of making a contribution by combining those different approaches. And so we view environmental health literacy as sort of the building blocks made up of 3 components that'll help individuals and communities combat environmental health risks that they face. The first component is knowing, right? So factual knowledge plays an important but not sole or only role in people's willingness to engage in protective behaviors. They have to know some facts, right? Just knowing the facts isn't going to be enough to get them to motivate to change, but it is an important aspect. The next thing that we identified as an important element is feeling, right? Being prepared to decide means that you feel like you have good information and that information exceeds your, what we call the sufficiency threshold, that you have a sufficient amount of information to make a good decision for yourself. And then the third component is believing. So thinking that the protective actions will work because they actually do work, thanks to science, right? So a good example of this is wearing masks during a global pandemic, right? If you want to convince somebody to wear a mask, they have to first believe that that's actually going to do something. So I said before, we're focusing on environmental health literacy in the people of North Carolina, and we had sort of a theoretically derived motivation for conducting this study in particular. We wanted to look at known factors underlying health disparities and see if incorporating environmental health literacy helped people overcome those disparities. So we looked in particular at socioeconomic status. So we know that people of lower socioeconomic status are more susceptible or have an increased susceptibility to adverse health outcomes in environmental health. And we also know that race and ethnicity has played a role historically in these risks. Historically marginalized communities have experienced, for example, environmental injustice or face unique problems based on historical factors that play a role. So how do these factors play into potential disparities in protective health behaviors? So we're identifying where we see disparities in health outcomes, and we're sort of going into, okay, how do we look at disparities in protective health behaviors? So our research expectations are basically that we're going to look at socioeconomic and or race and ethnicity disparities. We're going to incorporate environmental health literacy, and we're going to see if we have a reduction in disparities due to environmental health literacy. So the last component before I can share our results with you is what specific environmental health threats are we looking at? So as I said before, we're looking at the state of North Carolina. At the Center for Human Health and the Environment in our Community Engagement Corps, we've been conducting semi-annual, our intention is to do it biennially, every 2 years, not twice a year. And survey of North Carolina residents, right? So 1,500 North Carolina residents who represent the entire state and sort of take the temperature on how they feel about environmental health threats. So we've gotten some very interesting survey data. The data I'm presenting to you today is from 2020. If any of you are interested in knowing more about the data that we've collected so far and the health threats we've looked at, I'd be happy to talk with you. In 2020, we split our sample and we asked questions about COVID-19, which at the time was obviously very problematic. And we asked questions about PFAS, which you've heard about a number of times today. And so we approached these as distinct environmental health threats that might have different dynamics in terms of how things play out with environmental health literacy. I'll say one word on our research design and analysis. Again, we have 1,500 respondents from North Carolina that are representative of the state as a whole. And the way I approach this research is we built quantitative models to investigate the role of environmental health literacy. So we used ordinary least squares, multiple regressions. Our dependent variable is willingness to engage in protective behaviors, and our independent variables included, among other things, socioeconomic status, race, ethnicity, and our key intervening variable, environmental health literacy. So on this slide, I will be sharing with you the standardized regression coefficients. These are just part of our regression models. The whole regression models are reported on in the paper. And we basically divided up variables into different groups. So we have socioeconomic status, we have race and ethnicity, and we have environmental health literacy. And so for each of these variables, we have a stepwise regression model. So in model 1, we accounted for all the demographic factors. In model 2, we had demographic factors plus things like race and ethnicity. In model 4 is when we introduced the environmental health literacy component. So we're going to see how the things changed over time. And these are standardized regression coefficients, which many of you know are interpreted just like a correlation coefficient. So in terms of socioeconomic status and race and ethnicity on the topic of COVID-19, we found several significant predictors. We had level of education predicting people's willingness to engage in protective behaviors. If a person rented their home, they were more willing to engage in protective behaviors. And if the person was Black, they were more willing to engage in protective behaviors compared to white respondents. When we introduced environmental health literacy, those variables were no longer significant predictors of the willingness to engage in protective health behaviors, which tells us basically that the role of environmental health literacy is substantial in closing the gap when it comes to those particular factors in terms of disparities in people's willingness to engage. With PFAS, right, qualitatively a very different phenomenon from COVID-19, and so that's one reason we want to do this comparison. We had the same sort of setup, and here we found only one significant predictor of willingness to behave among these first elements of interest, and that was Black respondents, again, were more willing to engage in protective behaviors. And when we introduced environmental health literacy, we saw that that variable also became no longer significant. So it was mediated by factual knowledge and people's level of response efficacy. That's the believing component, that they believe that things will work. So what does this all mean in terms of 21st century solutions? Well, we saw little role for socioeconomic status, some evidence in the context of PFAS, but not consistent once EHL was added, and some role for race, but not for ethnicity. So Black respondents were more willing than white respondents to engage in protective behaviors, which in itself I think is an interesting phenomenon, considering that on health outcomes, Black participants and Black residents have worse health outcomes than white participants, right, on average. And so there's an interesting inversion of they're more willing to protect themselves, but they have worse health outcomes, and I'll speak to that in a minute. And then that difference between Black and white respondents disappeared when we took into account environmental health literacy. So coming back to this question, right, we found a disparity due to race, and one of the questions we asked ourselves is why, right? I think there's a couple reasons I could think of off the top of my head. There may be more, right? So Black residents of North Carolina are from a historically marginalized group, and those groups of people often have had to identify and act against threats to their communities and their health without institutional help, right? They can't necessarily rely on institutions that have traditionally been controlled by the majority to help them. And so that's one reason why they may have a higher level of individual willingness to protect themselves. Another reason, which has more to do with communication, is that marginalized groups must manage what's often referred to as co-cultural communication, right? In other words, they have to navigate their own culture and the majority culture, right? They have to have one foot in each of those cultures in order to exist in the world and to get things done. And I think that speaks to, it's a burden to have to do that, right? And I think that that speaks a little bit to why you might see this disparity. Of course, we also found that environmental health literacy reduced those disparities, and so the other question is, how did it do that? I think one reason is, or one way in which it did that was centering collective efforts against environmental health threats on communication around the 3 dimensions of EHL, right? So if we focus on these 3 dimensions of EHL, environmental health literacy, we can be sure to close the gap on some of these disparities in people's willingness to engage in protective health behaviors. We can encourage these elements in our communications with vulnerable communities in particular to encourage people to protect themselves. And we can help people make informed decisions, right? I think a lot of the presentations today have highlighted a lot of risks that we face in modern society. And unfortunately, the truth is that we can't live in a zero-risk society, right? We have to make choices. And for some people, being able to make informed choices is not something that they have access to in their everyday lives. And I think it's something we need to emphasize, that we want to help people make the best choices for them in terms of protecting their health. I'll speak briefly about some ongoing research we have at the Center for Human Health and the Environment. We focused on environmental health literacy, which is a key component of our work in community engagement. Of course, we also want to look at more closely at the spread of information and potentially misinformation in the state of North Carolina. We want to investigate these dimensions of literacy in other health contexts. And we'd like to engage in more qualitative research to sort of complement our population level quantitative analysis of this survey data. And with that, I would like to make several acknowledgments. First, to the Center for Human Health and the Environment for funding our survey research, which made this study possible. Thanks to my colleague Katie May, who is my partner in crime and the co-director of Community Engagement Corps. Thanks to our co-authors, and the article is available online in the International Journal of Environmental Research and Public Health. And I will say thank you. I worked in radio for a time, so it was like, oh no, 5 seconds left.
Do we have any questions?
I was just curious because the survey you did was beginning of the COVID pandemic, and there was a politically inspired movement to deny science, which may have affected other than the black community more, which would cause a difference. Mm-hmm. Did you look at that at all?
So that's a good question in terms of when, I should have said when we conducted our survey. So it was in June of 2020. And based on concurrent analysis I've done of social media commentary on COVID-19, primarily on Twitter, RIP, that was sort of right before the time that social media commentary peaked it was starting to decline at that point. And we didn't see as much misinformation yet in June of 2020 as there was to come later on. But I think absolutely, right, the way that COVID-19 impacted the black community in North Carolina certainly would have played a role in people's willingness to engage in protective behaviors, especially because, you know, at that time in the pandemic, we were just sort of all on our own, right? There was no kind of collective way to protect ourselves other than people making specific kinds of decisions. So, yeah, I mean, I think if you looked at it later on, and we have COVID-19 data from 2021 on our survey, that'd be very interesting to do a comparison to that I haven't done yet. But we also asked a question in that survey of, did you get vaccinated against COVID-19? And we asked people who said yes, why did you get vaccinated? We asked people who said no, why didn't you get vaccinated? And so we've got some qualitative data that I'd like to look at as well in terms of following up on that. Thank you all very much.
Okay, the This concludes our highlighted speakers for the afternoon. We have now, before we go to our lightning round, we have a panel. This panel is organized and chaired by Professor Aaron Sills. Aaron is a member of the Research Leadership Academy and the department head of Forestry and Environmental Resources. And Aaron, can you come up and introduce your panel?
Thanks very much, and good afternoon, everyone. So as Heike said, we're going to shift now from talking about literacy and responses to environmental health risks, sort of go through the looking glass and think about environmental health solutions. That is, how can we manage the environment and natural resources to provide solutions to 21st century threats to health. Right, so we're going to have 3 panelists. I'll ask them each to introduce themselves, and then we'll have plenty of time for question and answer and discussion. And this is a quiet group in here, so please gear yourselves up to ask lots of questions so we can have a dynamic panel. Let me go ahead and call the 3 panelists up, please. Any seat you want. We can do musical chairs.
Huh? Oh, I don't.
Great. So we'll get started here. I'll introduce each one and then let them tell you a little bit about themselves and their research and how it relates to environmental health solutions. So 2 or 3 minutes each. Actually, maybe I'll switch course. I'll introduce all 3 of them, and then I'll let them go down the line and give you their introductions. So starting on your left is Dr. Elizabeth Nichols, who is a professor in forestry and environmental resources in the College of Natural Resources. Then we have Dr. Aaron Hipp, who's a professor in parks, recreation, and tourism management, also in the College of Natural Resources. And Dr. Katie Martin, who's an assistant professor, also in forestry and environmental resources in the College of Natural Resources. So now you know who you've got on the platform up here, and I'll let each of them do a more thorough introduction of themselves and their research as relevant to environmental health solutions. So Elizabeth.
Or do you want me to do the visuals? All right. Nice to see y'all. Hi. I haven't seen you in a long time. I was trying to run. Okay, so I work at the interface between water quality, and forest systems in different ways, sometimes using forests to clean up contamination in the subsurface. More recently, looking at forests as they are being used in North Carolina as treatments for municipal wastewater systems. We're sort of different than our other sister southern states in that DEQ has been doing this since the early 90s. These are permitted US EPA wastewater treatment systems. They tend to be in rural areas. About half of them, I think there are about 86 right now that are municipal wastewater. About half of them are forest systems. Other systems might be growing alfalfa typically. And I want to look at this in context to one of the largest systems in the country, that is in Jacksonville, North Carolina. It is a forest wastewater treatment system for the city of Jacksonville. And here you're looking at Hurricane Florence as it approaches. And this is a system that is probably operating at 60% capacity, which is great because that area of the state, like here, is exploding with population and growth. And it's a forest system of mostly pine trees, but some mixed hardwood. And the wastewater comes in from everyone in the city and sort of some adjacent county folks. It sits for a little bit in these lagoons after sort of get the big stuff out of it, and then it's sprayed out onto the forest. So the forest itself normally sees about 50 to 60 inches of rain per year, but with the additional irrigation, it's doubled. So I got interested in these systems for a couple of reasons. We got some calls from some smaller forest water reclamation sites where they were having some problems. These were typically management issues around applying too much and not rotating it around. And we got involved with replanting the forest system itself. I was very interested in this system because it's so big. And one thing that I really wanted to know that you've already been exposed to, I guess, earlier today is what happens to all the chemicals in the wastewater as they go through this forest system. And I want you to understand these are permitted facilities, so they have to keep up with data regarding biological pathogens, and they have regulations around what they can and can't do. And they meet those when properly managed, and most of them are. But we really haven't looked at them with regards to emerging contaminants. So another interesting question I had is, how does all that water impact the forest productivity? So to give a really short spiel of that, these are the systems that exist on top that are land application systems I'm talking about. The conventional wastewater systems are in the lower map that you see. And I'm going to come to this in just a second. From the Jacksonville site, the trees, doesn't matter whether it's a lot of water that they're getting from irrigation or in the non-irrigated. It's a soil series that really grows and determines how well they grow. So there is no impact from the actual excess wastewater application. What I thought was really interesting for this site is it was a direct hit with Hurricane Florence, and so in a 24-hour period, it got 30 inches of rain. Most conventional wastewater treatment plants in that area failed due to flooding. This system never failed, and it was operational the next day. So they're very different systems in terms of the energy input requirements and the management requirements, but they also are systems that give us a lot of ecosystem services and benefits. What happens during drought, which is what I got funded to study, and then we had 5 years of too much rain. During drought, this system acts as basically a groundwater resource to adjacent agricultural irrigation systems around it. So the chemicals that are coming in and getting sprayed out that make it into the very shallow groundwater system do make their way off-site for use in agriculture, particularly under times of drought. In times of flood, and we are able to follow this system after Florence, those lagoons, and they're really, really huge lagoons, filled a lot with rainwater from Florence. They had to do excess spraying before Florence arrived to be able to handle that. So this system acted as a dilution mechanism for the rest of the watershed. Because now they had so much water from the rainwater, they still had to do excess irrigation. So PFAS and all sorts of other chemicals coming down upstream were diluted out by the system itself with regards to surface water concentrations. I thought that was pretty neat, and that was also true in the groundwater. And that continued for 6 more months. So that was a benefit that I never anticipated in seeing these types of systems, and just the fact that it maintained its functionality. All right, what you're looking at on this slide at the top there are lots of different land application systems. I just talked about the municipal wastewater. Some single families have these systems permitted for their septic systems. And then you have, of course, land-applied biosolids. The bottom slide is the above slide with where we're pulling water for municipal drinking water systems. And I wanted to show you that, just to show you the density, and for you to understand that we're already in the business of water reuse at various spatial levels and across the state. And it is one of the emerging issues or current issues with regard to climate change and what we're thinking about for environmental health. And groundwater, particularly on the coast, coastal communities like to have groundwater from the deep, deep old aquifers. We're finding that the chemicals that we're using are finding their way into those deep aquifers mainly because we are using them and making them sort of like Swiss cheese with more wells. And so we're creating conditions where we're exacerbating water reuse in terms of transmitting chemicals to places they normally would take longer to get to. And so this is already a part of the reality of what we do as we flush our toilets here, and then the people of Smithfield drink their tap water, right? So this is something that's just commonplace to our current world. And the big issue around that, in lots of respects, would be the chemicals. So this just gives you a look at some of the chemicals the EPA is concerned about. There are a lot of them. We've been screening for them in these different systems. And the forest water system is just a huge filtration device, but I think it's a very effective one and makes sense a lot from both its economic costs and its resiliency to flood and drought. And that's just a look at the system itself. It's outlined. It's about 7,200 acres. And that's just a brief review of what I talked about. I think these slides are in reverse. Yeah. Well, the last thing would be, how did the community view these things? Once they understood the function and how well it worked, they were actually quite pleased that they were receiving the service from the forest water reclamation site. And this was all part of understanding that they'd be more amenable to that if it was going on to food crops. And they had some really great recommendations for how they would be more acceptable to that with some additional research.
Thank you. So that starts us off with a great case study of an environmental health solution. Let's get our other two panelists to introduce themselves, the areas they work in, and then I'll be looking for your questions.
Yep. Hi, everyone. Again, Aaron Hipp. I'm with the Department of Parks, Recreation, Tourism Management, as well as the Center for Geospatial Analytics, and am part of the Global One Health Academy here. I don't have slides, but what I am going to ask you to do, because this is about solutions, environmental solutions to 21st century threats to health. Sedentary time is a huge threat to our health, so please stand up if you're able, if you're willing. We're not gonna dance, but feel free to dance, spin around, whatever. Stand up if you're able. Because I am in Parks and Recreation, I am interested in activity. I'm interested in how the environments we're in impact our everyday lives, how we move, how we talk with each other, how we socialize, our mental health. And so, the setup of conference rooms or classrooms are very much a part of our everyday lives. But towards, our environmental solutions and our nature-based solutions here and more relevant to today, and it builds great off of Elizabeth's work. You're welcome to stand the whole time. Not a problem, because I'm forced to, so you're welcome to as well. But yeah, so nature-based solutions, it comes off of some of what Elizabeth was just talking about and a graduate student that we worked with that looked at stormwater and how communities across the U.S. are designing their cities and designing their communities to capture the stormwater and these green spaces that they're using. So both the design of the green spaces, and are they, say, a kid's soccer field, are they a greenway, but then they can flood when you have flooding events, right? Or how is this water captured and how is this available? So this student was interested in this. This is Austin Thompson, a former PhD student who's over at UNC now. She was also really interested in how do you fund this? How do you work with cities to fund these? How do you encourage small rural communities in North Carolina or across the South to think about green-based solutions or nature-based solutions and to choose these? Because oftentimes they're more expensive up front, but they have what she came to term as slow capital. The city, the county, the town can regain a lot of those costs, but it's in a much longer time span of like 20, 25, 30 years. More like a house mortgage, right, than that instant impact. So that was one aspect. But to get kind of more into the work that I'm doing every day, we take this idea of access to spaces to recreate and spaces to be with other people and to have this psychological restoration. And so a really nice example of this, and I think a decent segue, is in Fair Bluffs, North Carolina. So this is a town in Columbus County, North Carolina, very close to the border of South Carolina, near the giant sombrero at south of the border. And so Fair Bluffs has flooded during hurricanes. And they're now moving this downtown up 6 feet elevation, about half a mile away from the river there. I believe it's the Lumber River. And so the downtown is going to be raised, and a park and a greenway are going to be built there. So this is going to be an amenity for the community that has stayed. The downtown is going to be rebuilt, again, at a 6 feet higher elevation. But yeah, so we're working with Fair Bluffs and with Columbus County right now through a CDC grant for high obesity, for their high obesity project. So these grants were only available to the land-grant institutions across the U.S. that had multiple counties with at least 40% obesity. North Carolina currently has 14 counties with at least 40% obesity. We're working with 10 of these counties, and we're looking at access to fruits and vegetables. We're working with the PhytoRx program that was developed here at NC State at the Kannapolis campus. And then we're working on everyday access to places to recreation. So how can we work with communities to have access to these spaces? And they're all rural communities. Robeson County, Columbus County, Bertie County. That's where I spent my spring break last week. But great, great communities. And it's really just emphasizing what they have available to them and making sure access is there. But access is just one piece, right? You've got to be able to get to these places. You have to have these places available to you. Another piece that we've worked on is called green social prescribing. And this was originally funded by the UGPN. And so it's an international collaboration with our colleagues in Australia and UK. And so the idea there is, how do we work with our primary care physicians, our nurses, our school nurses to prescribe access and use of green spaces to all ages? And we were interested in physical activity. We're, again, interested in social health, as well as psychological well-being. What is it that both potential patients are looking for, as well what are providers looking for, and that information exchange there? That led to some current work here on campus. As you know, we've had some struggles with student, staff, and faculty well-being over the past few years. So we are working both to make our buildings and our campus safer, but also to emphasize the myriad of green spaces that we have on campus. I saw Jude Desanoyer earlier and Activate Lake Raleigh and what they're doing on Centennial campus to activate the lake and the trails around the lake and accessing the greenway there. Nathan Williams is doing some great stuff. He has worked with Campus Recreation. He's currently a faculty member with us and is thinking about what's available to students, faculty, and staff, and then how also do you bring virtual reality into that conversation to get people to engage when they might not be able to get out to these spaces. And we're also adding to our campus map visualizations and augmented and virtual reality experiences so that students and parents can see what is available to our students here on campus in terms of green spaces. And then I'll end with 3 quick other related ideas. So one is with the National Recreation and Park Association. We're working with Robeson County again, as well as Mount Airy. And we're thinking about pathways to careers in outdoor recreation. And so how are we engaging with high school students who hopefully become NC State students to engage with them to encourage their communities to recreate outside and use these spaces. And then the last 2 examples are unique examples funded through the Robert Wood Johnson Foundation. One is right here in Raleigh. The Southeast Raleigh Elementary School was built and opened in 2019, and it shares a roof with a YMCA. And within this school and YMCA for elementary school, they have access to 3 kitchens where students can learn about cooking and fruits and vegetables. So they have a rooftop garden and a normal space garden where students can grow their own vegetables. They have access to a greenway, and then they have park space. And the new bus rapid transit will go through there. And so engaging how the students' families, and how the teachers who work there are engaging in these green spaces throughout the school year. They also have a great food distribution program there. And the last piece kind of comes back a little to campus and a little to that Southeast Raleigh, and that's thinking about where we live and multifamily developments. We worked with the Robert Wood Johnson Foundation and the College of Design here to look at how our multifamily developments, so you can think of the new sky-rises coming downtown, how they're using things like dog parks, rooftop gardens, access to parks, access to Dorothea Dix parks, how they're using that to sell their properties, to rent their properties, how they're using these nature-based solutions that our communities are providing to make money on the developments that they've had. And so I think to close out my comments, it's very interdisciplinary. It's very much working with communities as well, because it's going to be different in Fair Bluffs versus Southeast Raleigh. And so these strategies, we're facing a myriad of challenges. The strategies are often very local, but they take a lot of collaborations. So I will end there.
All right, good afternoon. I'll be succinct because I can tell that you have a lot of burning questions for the other panelists. Former Peace Corps volunteer as well, so just wanted to mention that. How does my research relate to environmental health solutions? So I spend most of my time thinking about climate change, so I kind of feel like that's a sort of an existential threat to our sustainability of the planet. So on a broad scale, everything I do relates to human health, right? Particularly vulnerable people who are in places that are going to be affected by more extreme events. In terms of more on-the-ground research, I work on things like increasing green space. So across both the natural and built environment, I'm an advocate of greening. And in our biome, I am an advocate of forests. Both in the built environment, we've worked on urban green spaces and trying to increase green infrastructure, which is kind of the traditional definition is for stormwater management in urban environments. And we do encourage that, but we look more broadly at green infrastructure for protecting source water for drinking water. So I've worked on green infrastructure in cities funded by the North Carolina Sea Grant, looking at green space on elementary school campuses, and looking at potential disparities in green space by free and reduced lunch and student demographics in terms of race and ethnicities. We did find, fortunately, that there are not the disparities on elementary school campuses that we do see in our neighborhoods, so this has good potential. We did see that the types of green space tends to be kind of open playing fields, which of course there's a lot of benefit to that, but we think we could really increase the tree canopy cover. There's something special about trees, not only because I'm coming from the forestry department, but also the research suggests that the benefits of trees are even greater than any other kind of, just kind of a field situation. I also work on a project with a group called Keeping Forests, which is a consortium of state agencies, timber products companies, conservation nonprofits to look at ways to maintain forest cover throughout the southeastern United States as we go into a future where the secret's out, it's a great place to live, so people are moving here, and as a result, we are turning a lot of our forested areas into urban development. But we do see that out of all of our different land cover types, forests are the most protective for our drinking water in particular. So anytime we convert forested areas to urban or agricultural lands, certainly there are other benefits that we see, but there's a trade-off in water quality. So we're looking at ways to maintain forest cover, incentivizing landowners, perhaps with some sort of economic benefit as we translate the avoided drinking water treatment costs into real dollars, but also looking at other ways throughout the landscape that we can use riparian buffers to protect water quality and protect from extreme events going forward. And so that's a pretty good summary. I will say I haven't been here all day today, and I apologize for missing some great information, but in my role here on campus, I feel like my best, most impactful force for environmental health solutions is teaching. So I teach a lot about climate change, I teach a lot about environmental justice, and I know that my papers are, hot off the press, people are clamoring for them. But when I have a captive audience of undergraduates, especially I teach an introductory environmental science course for students who are not science majors and who are only taking the class because it fulfills 2 GEPs,
I have that one semester with them that I can prepare them to go out and be a part of the solutions as citizens or professionals, whatever their role is in the environmental field, whether it's a passion or whether it's their job. I feel like that's my most impactful part of environmental solutions.
Thank you. So I hope that gave you 3 unique perspectives, expanding your solution set for the health challenges that we face, to include nature-based solutions, environmental solutions. I have some questions I could ask, but I want to pause here and see if those great presentations inspired any questions from the audience. Anyone want to start us off with a question out there? Taking Katie's cue about teaching, I'm going to pause and let the silence settle, and then someone will ask a question, perhaps. It worked. Thank you.
I think this would be for all of the panelists, but I'm wondering if you could tell us what you believe would be the biggest threats to green spaces locally in the next 5 years or so. I anticipate it's going to be an issue in the local elections in the fall, and I'm just interested in your take.
Thanks, Anthony. This one's on. Thanks. That's a good question. I think Katie hit on it some, right? Like just the development pressures here and what the strategies are. I think, like thinking about it locally, it's with some strong city governments in the triangle, and I don't see a really strong comprehensive triangle plan. I know there's a new Triangle Trails initiative, for example, that has some NC State experience on it. And so that's trying to engage 12 counties with greenways and trails, and I think there's some opportunities there. But there's a lot of challenges in terms of some different cities, even Wake County, having trying to limit growth and limit density within their own city limits, which is going to put pressure on other spaces. So I see that as a big challenge. I also think, there's tons of opportunities. We're just staring at one with Dorothea Dix right now. I do think the Triangle Trails Initiative is trying to do really nice things with connecting systems. An interesting pressure will be how cities and counties are able to work with developers on that choice of providing funds in lieu to help with some of the parks and green space master plans, or they're providing those spaces within the communities. And if they are providing them within these new developments, are those truly open and available to everyone? So that's my biased answer.
Sure. I have to say that I work a lot in interdisciplinary spaces. My core discipline is ecology. So first of all, people are new to the scene. And second of all, it depends is the answer to everything. But yeah, I see that the tension between, wanting to maintain green space and then, we do have some evidence that densifying cities is there's a lot of benefits, right? Reducing transportation and taking pressure off of other areas, providing different housing types, especially for affordable housing. So, working with students in particular, we've been talking about, can we develop differently? Like, can we have developers not clear all of the green space and then replant baby trees? Baby trees are good, but I have to, as many times as I can, I like to quote someone who was in charge of some of the quail hunting plantations, some of the best old growth in northern Florida. I would say it takes 500 years to grow a 500-year-old tree. So, just can we rethink the way we go about things and our relationship to the built environment? Just coming from a perspective where I often partner with social scientists, I wouldn't claim to have expertise in that, which is so critical.
Another question out there anywhere? Yes. I'll repeat that for folks online. So a lot of these solutions require scalability. Is that a hindrance?
I mean, I think that's a nice question. I think I see that as an opportunity that, and again, I mean, I think it's on each of us at a land-grant institution to realize the good work that's going on across North Carolina and our communities and take what is working and to understand the scalability. What pieces are scalable? Where are they scalable? With who are they scalable? I think there's opportunity there, right? Like there's challenges, and it's going to take some work and time and funding, right? But I think those locally known answers, usually there's a reason behind it, right? And so like what pieces of it can you take and scale across North Carolina, across the world? So I see it as an opportunity born out of that challenge.
Definitely, one size doesn't fit all. It depends. Like I live in Garner, and we have very lax tree rules compared to Cary, and the development is pretty impressive about clearing and repacking. And, you know, there are a lot of great initiatives in the state to rethink how we build and regrade sites and how we can get away from using stormwater retention ponds and have things that really are more nature-based. But to do that, you really would look at the original landscape for what makes sense, at least hydrologically. So I think you just have to have local activism. And you would know better than I, but there's a lot of research about how important it is for people to have access to green spaces, and I think everyone wants it near them. And so I think that's a great motivator, at least that I see in Garter in my involvement with them on green spaces.
Run with that question about scaling and ask about the other types of green solutions or green infrastructure we're talking about, whether it's using forests to provide clean drinking water or to manage wastes. Comments on whether those are difficult to scale, or maybe one of their beneficial attributes is that they're actually easy to scale.
I'll make the first comment, which is, and you might know more than I, Erin, about the Sentinel project. When you're really starting to go big scale, you've got to deal with the people who own the land, who are mostly individuals. So there was a military project very interested in keeping dark spaces, particularly on the coast, for military training. And that was an incentivization program for agricultural landowners not to sell out and create light spaces. And so I think you have to have that coordinated government-private partnership to do those things.
Other types of green infrastructure. Yeah, I would echo Dr. Nichols' comment that we in the eastern United States are reliant on private landowners and maintaining relationships with them because we have so much of our landscape is held in private parcels. Just for like scalability, I just wanted to provide a good news story in that I had a graduate student who worked on urban heat and the continuity of urban tree canopy cover. So we know for sure that more trees is better, but we were looking at kind of the configuration of trees and urban heat across 3 different cities and comparing those to indices of vulnerability of the populations. And we found that contiguous urban cover could reduce maximum July temperatures by 4 degrees Celsius, which is actually more than I thought. That's quite a lot. And that across Raleigh, Seattle, and Baltimore, there were only a few census tracts that were both devoid of tree cover and really high vulnerability of populations. So if we could start there, we could reduce their exposure to urban heat relatively quickly. Now, how difficult that is for a city government to address, I don't know, but it was a small percentage of the census tracts in each one. So that seemed like maybe something that we could start on.
Yes, I'm curious in thinking about kind of incentives and how to communicate the benefits of these green spaces, particularly in urban areas. Are people working on kind of ecosystem service evaluation specifically in urban areas? And I know ecosystem services, that's kind of, there are advocates and people that are not such fans, but might that be a way to kind of communicate these holistic benefits of like flooding mitigation, mental health benefits in a way that would help developers avoid clear cutting, for example, if the city can understand here's the value of that in an economic sense?
Do you think about ecosystem services as mental health?
I do, yeah.
I haven't, but now I do.
Yeah.
Yeah.
Right.
A lot.
Yeah, I personally haven't done a ton with ecosystem services. Just a couple of graduate students have kind of gotten more into ecosystem services. So it's, yeah, I mean, it came up a little with the nature-based solution with Austin Thompson's PhD and looking at the stormwater, like nature-based solutions to stormwater. And so it came up there because, I mean, she was first interested in how the city can save money. And right, and some of that, you know, as Elizabeth was showing, that like with stormwater, you shut down your wastewater system if it overflows, and you've got really big problems for the city and for human health, et cetera. So there are, you know, I mean, you've got heat, you've got mental health, you've got stormwater and wastewater, and a lot of that can be tied into a single system. I don't know how well it is sold and communicated and how well you can put price tags on different things. I mean, I think that's kind of where some of the research is, to try and put prices on each of these aspects. But it's a good question.
The moderator is an economist, so. I'll say I think a lot about ecosystem services. I think I mean, in my interpretation, the whole goal of ecosystem services was try to put a value on nature because it wasn't being valued before. And I don't know how, I mean, we have potentially a ways to go, but perhaps the moderator has a response to that.
Okay, so how can I not jump into that? There are some of the urban forestry tools that the Forest Service have developed that have monetary modules built onto them to try to quantify some of these services of urban trees in particular. So I think one opportunity there is to think more broadly about the services provided by various types of urban green space, in addition to trees, which is usually what the urban forestry department is responsible for. Okay, next question.
Can I just a real quick fun story, I think, somewhat related to that. I don't know why I'm thinking about wine right now, but I am. But so in the Center for Geospatial Analytics, they're doing a really cool study right now with Google Street View, and they're automated the process of what type of trees are along paths. And so they know that like this certain type of pest in vineyards in California, it's going to transfer from one vineyard to another via the street trees along the way. And they're now trying to figure out between the different vineyards in California which are most susceptible to this transfer based on these pests preferring certain types of trees. And so that's another way to look at forestry from a very different lens. Yeah, so just an interesting thing that's going on campus.
Okay, so let me give you another prompt to think about here. So I think it's a nice example for thinking about the wide range of types of nature-based solutions there. You might think that there's a solution in having a more diverse urban forest, right, to provide a barrier to transmission of pests and sort of environmental health and even agricultural crop health. So I think we've got lots of fantastic ideas here, but I want to sort of harken back to what Francis de los Reyes was talking about when he said that we often don't think about how waste is processed because that infrastructure is hidden, it's underground. I think we have a slightly different challenge here, which is nature is all around us, and it seems like it always has been here and always will. And so but in a sort of the same way, then we don't think about it as a specific solution, as a potential solution out there for some of our public health threats that we face. So what I wanted to ask the panel is to sort of think broadly about the wide set of possible environmental or nature-based solutions, and imagine yourself in an elevator ride, perhaps a long elevator ride with like the director of NIH or the Gates Foundation. What's the key? What do we need in order to move forward on these environmental health solutions? Maybe it's monetizing ecosystem services. Maybe you've got some other ideas.
I'm in the elevator first.
Yes.
Go for it.
We were prompted about this question, and I have been giving it some thought, and I'm not sure. I don't quite have my pitch yet for the Gates Foundation. And here's why. Because when you create green spaces, and particularly if it's like these forest reclamation sites, people are not going to be on site. No one likes to get sprayed with wastewater, right? But they still have tremendous value in and of themselves from an ecosystem point of view, just because there aren't a lot of humans on it. They're not the risky sites that I think people would initially perceive them to be because of wastewater and, E. coli and things of that nature. And I always kind of chuckle when I see all these stormwater retention ponds and all the Canadian geese that live within the fences because it protects their babies so that we have even more Canadian geese. And there's lots of poop that come out of those geese. So one of the challenges with nature-based solutions is you have to accept what comes with nature, which are often things that we're measuring to avoid. So I don't quite have my pitch ready.
Okay, let's go on to the next pitch. So we've got a complication. What else do we want to tell NIH or the Gates Foundation?
He's going to say, take the stairs.
Yeah, take the stairs and have a glass of red wine. No, I mean, I think it's the multi-benefits. It's that you can benefit nature, you can benefit biodiversity, you can support different biomes, but that you can support human health in these spaces. And then, a huge piece of this is just equitable access and the communications of who has access to these spaces and how they're designed. And so I think that's where I would go with that. There's multiple benefits. We need to focus on making sure that everyone has access to these multiple benefits.
Yeah, I've been working over my claustrophobia in elevators. I was too scared of stairs for health and because I don't like small spaces. Yeah, I'd say, you could go radical and ask them to come outside with you and just see what they hear, what they smell, what they feel when you're out in nature. Because you can take it for granted. It is all around here in Raleigh. Now we have one of the highest urban tree canopy covers. But it's changing rapidly. And we want good places for people to live and work. I try to emphasize to my graduate students when speaking about land cover change, we don't want to negate the fact that, we live in the city and we benefit from all the amenities there. But yeah, I guess I would ask these people who are, the titans of society, how do we, the benefits of nature are public, but the consequences of environmental trade-offs are also usually public, and the profits are usually private. So how do we go about, I don't know if payment for ecosystem services is the right way, but how do we go about increasing the value in a way that is motivational of all the benefits that we get from nature, and how do we share in the burdens of the trade-offs of a modern society?
So we're down to a couple minutes. Anyone have a burning question? If not, I think I'll give each panelist, say, 30 seconds for final words. I'll give you a little nudge if your final words want to be about an issue we've been talking around a little bit, which is about equity and whether these solutions are actually accessible to marginalized or traditionally underserved communities. So either that or whatever other final words you'd like to offer to us on environmental solutions.
I think my final words around equity and these issues is that people value nature in different ways. And if we were all in this room going to plan a green space, we would have different values about what we would want that green space to look like. And not everyone wants to be next to a green space that has raccoons and foxes. And so it's the work of sociologists and such like that to help manage those values and outcomes.
Yeah, I think that's a great point. So, spatially, geographically, access is often equal and sometimes greater in under-resourced communities. But the quality of those spaces, the programming that's there, the sense of belonging and inclusion that is there is often less. And so I think that builds well off of Elizabeth's point.
Yeah, I would say it seems to me, which is kind of an interesting statement coming from an ecologist, access to participation in the conversation is key to equity. That the past has been shaped by a select number of voices, and to have a great future, I think we have to make room in the conversation.
Thank you for a stimulating conversation. Let's give them a round of applause.
Thank you, everyone. Thank you, our panelists and our moderator. I love hearing the different contributions we have here at NC State. What we are going to do, since we're a little late into our agenda, we're going to take 3 to 5 minutes to organize the second faculty round. So if you are a faculty member participating in the second round of lightning round presentations, please meet Sophia Colon. She's waving her hand now by this table that says reserve close to the break service. We're just going to take a couple minutes to organize that session of presenters, and we're going to bring them back up. So we're going to move fairly quickly into our next agenda item here. But if you do have to go grab a drink of water or a quick bio break, this is your chance. And then we will move from there directly into a dessert social. So if you, again, are a member, or if you're a participant in this second round of faculty lightning round presentations, please meet near the break service station in the back, and we'll be up shortly to start the next section. Thank you. presentations of some amazing research that's happening here at NC State. And some of the items, this agenda item provides our faculty a chance to showcase their research and provide a platform for the NC State community to discover research across campus. Presenters will have 2 minutes to present their research or scholarship as relevant to any aspect of the 21st century threats to health. First up, we are going to ask Max Scott to come to the stage.
Okay. Right, thank you. My name is Max Scott. I'm from the Entomology and Plant Pathology Department. And I'll give you a quick introduction to some of the things we do. So this is a CRISPR-based gene drive. Maybe that pointer doesn't work. Okay, so in this example, what we've done is we've inserted a gene that expresses Cas9 and a guide RNA into a female essential gene. We did that before we started this. And so what happens, this is expressed in the germline, and the Cas9 guide RNA complex cuts the female essential gene, and through homology-dependent repair, gets copied across and disrupts the female essential gene. So this happens in the germline of males, and so this male has gone from a heterozygote to a homozygote, so all the sperm are producing, carry this female disrupted gene. And this sort of diagrams what happens if you release these males, they mate with wild-type females. In this case, the disrupted female gene is essential so that all the female offspring are sterile. The male offspring are fine, and the gene drive works again in the germline so that all the sperm carry this disrupted female gene. They mate with a fertile female, and again, all the female offspring are sterile, and the male are fertile and the gene drive works again. So this is a way of controlling populations. It can be very effective, and it is the insect itself, the pest, that is the control agent. So we call this a fully autonomous drive. And we work with mathematicians. So we work with Alan Lloyd and his student Cole Butler. So we made a split drive where Cas9's on a separate chromosome. So the modeling suggested that within a few generations we could eradicate a cage population. And we tested this in spotted winged Drosophila, an invasive fruit fly that was first found in North Carolina in 2010. It attacks berries. It's a pretty dangerous pest. And so in these small cage experiments, you can see the gene drive cages, we have almost successfully eradicated the population after 9 generations. So this was quite a big deal for our lab. This was the first successful gene drive developed for an agricultural pest, and some of it was published last year. Time's up. Thank you.
Hello, everyone. Thank you for joining me today. My name is Ian Williamson. I am from the MBS department in the veterinary school, and I study the gut, and in particular, I study enterocytes, which are these cells that line the internal lumen of the gut. So when you cut the cross-section of the intestine as a cylinder, it is dominated by these microstructures which protrude into this luminal space. The cells that line that lumen are called enterocytes, and I use animal and organoid models to study their function. Organoids are a culture system where we can remove these cells from the body and grow them in a hydrogel, kind of like Jell-O. And I study enterocytes for their properties in whole body metabolism, and mainly through their ability to convert nutrients from one form to another. This was kind of a seminal paper during my training where other group discovered that the fructose component of sucrose, which is a disaccharide of fructose and glucose, is converted by these cells into glucose during this absorptive process. And being that glucose elicits insulin signaling and fructose does not, this had very profound potential for augmenting whole body health. And they went on to show that either through dietary mechanisms or genetic ablation, you can increase or decrease the ability of the intestine to convert nutrients and have profound effects on downstream responses, driving things like fatty liver diseases. And so my lab uses microbial and dietary modifications in order to augment enterocyte nutrient conversion in order to benefit and reduce symptomatic presence in various disease models. I have 10 seconds left. Go for it.
Hi, everyone. My name's Acer Van Wallendale. I'm a new professor in horticulture and crop and soil science. I'm in the weed science program, and my research program focuses on weeds that are closely related to crop species. So if you've ever seen a weed growing out of a tiny crack in the sidewalk and wondered why are we not using this weed to grow our crops, I'm going to be the one who's trying to answer that question. So in the phase of climate change, we know that weeds are stress tolerant, quick growing, and diverse. So I want to answer, what can studying weeds teach us about growing under climate change? And this is a weed that I studied in my graduate work and forgot about in a Coleman cooler for about a month, and it managed to escape. That one doesn't taste very good, but we have some other ones. One of the main species I'm studying is called proso millet. It is thought to be the most drought-tolerant grain crop that there is, but it's also very interesting because there are populations that grow as agricultural weeds around the world, and they look very different. What I've been able to do is study the genetic differences between these groups using whole genome sequencing and find genes that are completely different between the groups and are related to traits that are involved in both weediness and domestication, like this grain number gene, GN1A here. And I've also been able to look at hybrids that we found in the environment and am looking into potentially making crosses between these to introduce the stress tolerance into crop populations. Oh, thank you very much.
Hi, everybody. My name is Fikret Isik. I co-manage a tree improvement program in the Department of Forestry and Environmental Resources. The tree improvement program supports the big pine industry in the southern United States. One of our research priority is to understand the fusiform rust fungus and pine gene interactions. This is the major pathogen infecting pines, pine plantations, causing a lot of damage. The pathogen is endemic, biotropic species, and it goes through life cycle and then infects pines and oaks. So the research focused on discovery of those fusiform rust genes in pine. And we discovered 2 genes on linkage group 2. And the seedling genotypes inheriting 2 genes, they are almost immune to disease. Those 2 genes are linked in repulsions on linkage group 2. So the research project we are currently working on is trying to understand the virulence in the fungal populations, how those single spore isolates and the virulence among those single spore isolates overcoming those pine genes, and how do they interact at the molecular level. So one of the objectives of the research is to sequence single spore isolates of those fungus populations and then develop a pangenome of the fungus and map those virulence genes in the fungal genome.
Good afternoon. I'm Jane Hoppin. I'm a professor in the Department of Biological Sciences and part of the Center for Human Health and the Environment. And I'm going to talk today about our work focusing on PFAS. These are chemicals that Sue Fenton talked about this morning about contamination of drinking water here in North Carolina. So in June of 2017, people in Wilmington woke up to find out they were drinking Teflon for 40 years. And so at a community meeting, they wanted some answers. And so we wrote a grant that we collected blood from 310 people 2 weeks after it was funded. And then we tested blood for 44 different PFAS, including some of the new PFAS that were related exclusively to the Chemours facility, including Gen X, which not only is a new chemical used, but it's also a byproduct of chemical production. And while we didn't identify Gen X in people's blood, we did measure 3 brand new chemicals in the blood of every person in Wilmington. And these are bulky chemicals that have environmental or biological half-lives of over a year. And there was no human health data available. And one of the key parts of our study is that the community wants to know what we know when we know it, even if we don't know what it means. So report back is a key part of that, and Andy Binder has helped us with some of that. And so what we also showed was that light blue are the brand new chemicals related to the Chemours facility, and dark blue are the PFAS that are commonly measured in everyone. And the far end is the national data. So you can see that overall, the levels of PFAS in the blood of people both in Wilmington, who are downriver, and people in Fayetteville who live around the plant are much higher than the US population. And now I have to go. So now we've built a cohort of over 1,000 people, 3 parts, Pittsburgh, Pittsburgh, Fayetteville, and Wilmington. And what we see is that Pittsburgh has the highest levels. Pittsburgh are the green, the highest levels of these chemicals, but all communities have much higher levels than the national average. And I'll thank you. Okay.
Good afternoon, everyone. My name is Deepthi Salvi. I'm faculty at Department of Food Bioprocessing and Nutritional Sciences at CALS. I'll be talking about preventing microbial contamination in food. We all know that there are different foodborne illnesses. In the US, 1 in 6 American citizen gets sick every year, and there is a lot of hospitalization and deaths associated with foodborne illnesses. Our lab works with different novel processing technologies which do not involve chemical residue. And those technologies include cold plasma, which is the 4th state of matter. We use that for inactivation of pathogenic microorganisms. High pressure processing, where we use extremely high pressure, more than the pressures found at the bottom of ocean, to inactivate the bacteria, or UV lights to make food and food contact surfaces safe. Some of the plasma projects include inactivation of bacteria or bacterial biofilms. These are pathogens on different surfaces, such as food and food contact surfaces, and then some of the in-package sanitation of the fresh produce. These are some of the projects that we work on. Some other projects that we work are on UV light, using 254 nanometer UV light and antimicrobial coating, as well as far UV light, which is a newer wavelength, which is much more safer for humans for inactivation of pathogens in powdered foods and also on the surfaces. Another research area that I'm interested in working with, and I will be seeking collaboration in, is engineering food engineering food to change its digestion in human gastrointestinal tract to impact human health. So these are my different areas, and I'll be happy to collaborate. Thank you for the opportunity.
Hello, good afternoon. My name is Thomas Theis. I'm an associate professor in the chemistry department. I lead the NC State Hyperpolarization Lab, and our work is focused on developing new molecular imaging technology. We're at the intersection of chemistry, engineering, and spin physics, and our goal is to develop a new molecular imaging technology that is truly scalable and can be available for everybody. So to put that into perspective, today's medical imaging is dominated by very costly devices such as positron emission tomography or MRI instruments. And with our hyperpolarization technology, we're trying to establish a technology that can give us boosts in sensitivity several millionfold, so that then we can now work with very low magnetic fields, very inexpensive devices. And so we combine the idea of using the entangled quantum state of hydrogen gas, and we develop the chemistries to transfer this polarization onto metabolites and drugs, vitamins, that we can now use as contrast agents in MRI instrumentation. So the bottom picture illustrates the effect where a hyperpolarized agent is injected into a person with prostate cancer, and now we can actually monitor the chemistry of these molecules at low concentrations. So instead of just being able to look at the water in the MRI image, we can see the chemistry, and we have the potential to do this with low-cost instrumentation and at low magnetic fields. And because we are, of course, here also very interested in plant metabolism, I also want to say that the same technology can be used also to monitor metabolism, for example, in unperturbed soil. And we can look at metabolic turnover, for example, with devices that we can just bring to the field and do the analysis. Thank you.
Okay. Okay, yeah, my name is Yuanzu from Mechanical Aerospace Engineering. So what I'm going to show essentially is soft wearable sensor systems. So the keywords, well, soft wearables. For instance, we're using nanomaterials, and then we formulate them into inks, and then we print this on flexible, stretchable substrate, and then use for human wearables, also for plant wearables more recently. And then some other applications as well, for instance, sports, and then robotics. But to make those, we also using so-called sustainable nanomanufacturing, including recycling of some of the nanomaterials and also biodegradable materials. So here are just some samples of a range of the sensors we developed, like a strain sensor, for instance. It can detect as small as a pulse, okay? And then for blood pressure measurements, and also for lower back deformation, temperature sensor, pressure sensor, and hydration sensor, electronic textiles, from electrophysiological sensors to heaters, wearable heaters, okay? And then wearable plant sensors. And then some manufacturing, we can make a curve, the three-dimensional structures, and some integration issues. Okay. So here, this is a human wearable devices. Well, we have, I think the movie is not working, but anyway. So we have electronics. Let's see if it can work. Okay. So you can see, when the finger burns, you can see, we can collect in the real time the strain signals on your phone. Okay. Also, we have this plant wearable sensors, right? So all this work has done through collaboration with many folks on campus, and notably there's wearable sensors, Professor Qin Shenwei from chemical engineering. And we have some existing collaboration with folks from College of Natural Science and so on. So, okay, we're looking for more collaborations, biodegradable materials, human wearables for disease detection, rehabilitation, and some other applications possibly, and plant wearables to detect plant antibiotic stresses. And we haven't really worked on animals, so the sensors, we have used it for humans and plants, so probably the next one is animals. Thank you very much.
Yeah, hi, I'm Warren Jasper. I'm from the College of Textiles, and I'm going to talk about something a little bit different. We've heard a lot about the problem of drug resistance and combating pathogens. There's other some problems with pharmacological approaches to this. This is a very much a non-pharmacological approach. This is the idea of taking, let's say, a fabric and embedding electrodes in them and then generating a plasma on the surface. And what you can see here on the bottom, let's see, is this pointer? Yeah. This bottom one, this is with no voltage. And then when you apply a voltage, this is the filtration efficiency, and it sort of bumps up to HEPA levels. And you can do this very scalable. So like as an example, the nice thing about textiles is you can make very large devices with them. This is an example of a plasma on a couple of surfaces. The problem with filtration that we've heard about, like PPE, is it's really, don't think of it as a protection, but more like a focus. If you take all the air in this room and you just focus it down in through the filter, then you've got all this, all the pathogens right in a very small area. And then when you doff them off or you change something, then you can, you just re-aerosolize them. So that's not what you want to do. And so we looked at, let's see, yeah, here. I also am working with a colleague in College of Science, Frank Scholl, and we're looking at not just capturing the viruses, but also deactivating them. And so this is just an example. We can get about a 2 to 3 log reduction in viability by actually just generating these low-cost plasma filters. So thank you very much for your time.
Okay. And that almost concludes, the formal speaking session of our this year's University Research Symposium. Thank you, everybody, for coming. I don't know about you, but I made myself a little list. There were several, especially of these short lightning talks, where I wish we had more time, but I'm sure I will reach out to some of you. And I hope each of you has that little short list of where we need to have more contact. Right now, we will, Laura has prepared there's a survey, right, that goes out tomorrow or next week or whenever. Yeah, so we would appreciate if you fill it out so we know how we can improve or change it for next year. I want to really thank again Laura here and Sophia. Where's Sophia? There. For really handling everything in the background. It would not work without you. And I hear there's dessert, so please take the time, mingle a little bit more, and thank you, everybody who participated this year.